Hello, everyone. Welcome back to Ask a Scientist Gaming, where we combine mediocre gameplay with expert science. I'm Ken Hansen, Associate Professor in the Department of Chemistry and Biochemistry at Florida State University. I'm a photochemist/photophysicist. I care a lot about shining light on molecules and materials and tracking that light energy, whether it's breaking bonds or transferring electrons or moving energy around, hopefully doing something useful like solar energy conversion or catalysis for photomechanics. But more importantly, joining me today is Dr. Ashwanth Francis. Francis, if you wouldn't mind introducing yourself.
Hi, everyone. This is my first time on this stream, and so excited to be here. I'm an assistant professor at Florida State University. We study how viruses infect cells. Specifically, we label viruses and see them become fluorescent and watch them infect a cell. We track these particles and try to understand what the hell is going on and how we can block them from the infection cycle.
What do you typically teach?
I teach this section called Cell Structure and Function, and I'm hoping some of my students are there online right now.
Maybe.
Yeah, they're
anonymous. We'll find out.
Okay, they're a fun group of students, and I really like teaching the topic.
That's undergrad or grad?
That's for undergrads.
Okay.
And I also do teach graduate course for grad students on microscopy and bioimage analysis, which is what we do for our research as well. Yeah.
Nice. All right. Equally important. What game are we starting with tonight?
I guess we're looking at FIFA 2005 soccer. So that's going to be interesting. Yeah. I wonder if they had Schilacci and stuff, the Italian team. We find out.
They have a lot of teams. I was surprised. So go ahead and press start. It'll get you to the menu options.
Okay.
Start with this guy.
All right.
So you've played FIFA Soccer before, but not necessarily this version.
Yes, and it has been a very long time since I touched a joystick. Probably in the late 90s or early, maybe 2001. That's the last I remember.
Perfect.
Yeah. So let's do
Play
this.
now. X button is this
X
one.
button. Okay.
So yeah, we're doing PlayStation 1. This is FIFA 2005. Came out in 2004 because that's how these games work. But yeah, if you scroll to the right, this joystick here, there's a lot of leagues.
Spanish Primera.
You guys will have to let me know if the volume's too loud or not.
Italian Serie A. There you go.
Okay, so press down or press X. Oh, no. Try X again.
Okay, X again.
Oh,
Okay,
no,
I'll
you
go
gotta
back.
scroll through.
Yeah.
So when you pick the league, press X, and then you get to pick the team.
All right. Italia
Okay.
and AC Milan.
And these are your teams. These are the ones you cheer for.
Yes, so I studied in Italy for a very long time, and but before I went to Italy, I was still very I used to love Italian soccer actually. Inter Milan, let's do it.
Oh, who are you gonna play against?
Are you choosing someone?
No. It's you against the computer, but you get to pick their team.
Oh, is that right? Okay.
Avrilla, thank you for the follow. Thank you for joining us. Ask a Scientist Gaming, mediocre gameplay, expert science. We're playing some soccer tonight, ladies and gentlemen. Francis is picking.
Rest of the world. AC Sparta Prague.
Yeah, pick your worst enemy or the worst team.
Yeah, I know. FC Porto. They're pretty good, actually. Yeah. Okay.
I mean, who cares? I guess you get to pick the jersey if you want to.
Oh, should we have taken a different color?
Okay, so you want, move the joystick over to the team you want. Which one are you? Left, that one.
that.
Yep.
That's the one I want to be.
Okay, so X select. There's your team. Do you
All
recognize the
right,
names?
Cannavaro, yes. Cordoba, Materazzi.
2005, it's been a while.
Zanetti. All right. Okay, let's go.
All right, press. It's loading. Are you ready for this? Do you have pride? Do you feel like you have to win now?
Oh, yes. Because I did already warn my students that I'm not going to be that great.
I mean, soccer's a good choice, though, right? Because the game kind of runs itself to some degree.
Yeah.
ASL, first time chat, thank you for joining us. This is my kind of game right here. I mean, yeah, it's pretty classic. I actually learned the rules of soccer from playing FIFA Soccer. So you're going to be the one... where are you?
Cannavaro. Okay, go for it. Oh, no.
Picks up the free ball.
Yeah, so you're going to be the yellow triangle. And I think you can do like tackles depending on the
Nice.
button, like a square or something like that. So I think X is pass, circle's going to be shoot. Or somebody that's a FIFA aficionado. I don't play a lot of it. I played it on the Sega way back in the day, but not on PlayStation. We're playing on PlayStation 1 right now. So if you guys have any suggestions, Francis is happy to take them while playing this game for the first time. And how long's it been?
It's been about 20 years now, I guess. Yes.
That's amazing.
Oh, man. I can't even, I don't even know.
So if you press the X button, I think it changes, which character you are.
Oh, no, he just scored a goal on me.
Oh, man. We're off to a start, ladies and gentlemen.
All right. It was all open.
Liliana, thank you for the follow. Thank you for joining us. Ask a Scientist Gaming. Mediocre gameplay. That's what we've seen.
And away we go, 1-0.
Yeah, I think so X button is going to change the character, so you don't want to hit that one too much. Yeah, the bottom button. Square is going to be is how you're going to, I think, tackle.
I see, okay. All right. Square is tackle.
Yep, there you go. Oh, no.
Red flag.
Full tackle. That's awesome. Oh, wow. So they're not offsides? What is going on here? Don't worry, you'll fall into a groove pretty quickly.
All righty.
As soon as you learn the controls, it's going to go great.
You have to be careful near the penalty area or else you can easily concede goals like that. They'll feel a bit more comfortable now. It's 2-0.
Probably should have picked different jersey colors, huh?
Yeah, that's what.
So did you play soccer?
I do play soccer.
You do?
Yeah,
You still
yeah,
play?
I love playing soccer, yeah.
Mm.
Oh no, I kicked it too far. Just not able to play soccer with my fingers.
Yeah.
Let's put it that way.
You do like team-based, club-based? Do you have,
I play with any team, anybody who's willing to play with me. Let's put it that way.
Nice. As long as they're willing to take you, it's perfect.
So soccer is one of those games I've always played without break, you know. So when I went to Italy, what happened was I started playing American football in Italy. I played for the American football team for Pisa University for some time. And then we got, let me get this guy. Oh, there you go. Oh, no.
Oh, nice. What?
That's a yellow card.
Oh.
Cannavaro. Jeez. So yeah, I played for the Pisa team. How do you pause this?
Start button here.
I see. Okay.
There you go.
So I played for the Pisa team for 2 years, and then we moved to Trento, and Trento is in the north of Italy, northeast of Italy. It's my favorite place on this planet, actually.
Awesome.
It's a very beautiful town, and there I found a bunch of grad students, and we used to work all the time in the lab. But Mondays we would not fail to go and play soccer. We played like with faculty and undergrads and grad students, all of that mixed together. It's a good culture, I would say, and we didn't miss the soccer game. Even if it rained or snowed,
Awesome.
That was awesome. Yeah, we totally loved it. And after that, when I moved back to India for a while, and then I moved to the US, I found a team at Emory School of Medicine. There were a bunch of students playing, and some of them worked with me, and so I went and played with them for quite some time. And then I've just not found time to play soccer. Really have to. I'm looking for a team in Tallahassee
I see.
to go and play with. Yeah.
I'm sure they have club of some kind.
Yeah.
But yeah, that's good to have that outlet for any grad students in the audience. Like, even undergrads too, just for like physical well-being, for mental health. Like,
Absolutely.
Yeah.
It's a great game for to play as a team and for to make a team, etc. Right?
Not too much physical damage or long-term CET.
Yeah,
unless you
that's
do a lot
right.
of headers.
That's right. You know, one of the reasons I stopped playing American football was I was in the defense, and I played with, I played as a linebacker, right? And there was once when I actually, oh, there's another yellow card. Just crashed in, and I think I sprained my back or something. I said,
Oof.
okay, that was fun. But I really enjoyed playing American football. It's so much fun, yeah.
Yeah, that was my sport back in high school.
Oh, is that right? Did you play football? So which position did you play?
I was a wide receiver and safety.
Oh, nice. So you got the height and
Yeah.
speed for that. That's right.
Yeah, I wasn't big enough. I was like hurt myself a lot, but yeah, like we won state. It was like, yeah, no, it was good. Lori's asking, what is our drink of choice tonight?
Oh,
So we are drinking, you said proof and IPA, so I went with La La Land.
La La Land. That's a great place.
So-
Yeah.
so
Cheers
Francis.
to that. Yeah.
Lori, cheers to you. We're drinking local. This company that's out of Tallahassee, Proof Brewing, has a nice brewery, and it has a bar in the student center as well.
I know.
But yeah, we're drinking La La Land. This is 7%. You guys can see the link there on Beer Advocate if you want to check it out. But hopefully you guys have a drink in hand. Celebrating on a Wednesday night. You taught today, so you don't teach tomorrow.
Yes, that's right.
So yeah, Lori, who just chatted, is our graduate advisor.
Oh,
So
nice.
she and I work together on
I see.
admissions, and we got our final count on graduate students this cycle.
Oh, wow. How many was there?
62.
Nice. New intakes?
Yes.
Oh, that's fantastic.
We were aiming for 45, and we got 62.
Wow.
Yeah.
Chemistry actually gets pretty good students. I've always been impressed by the talent. Oh man, how do I dribble?
Dribble?
Yeah.
I think you just run with it, right?
just run with it?
Yeah. Just don't
Just
press
keep going in
pass.
front, okay,
Yeah.
okay.
I think it'll automatically dribble to get some transport.
Oh, he took it. Oh yeah, so I just need to run without kicking. Got it.
Yeah. It's going to be a balance of controlling and passing, I guess, like real soccer, right? Speaking of real soccer, have you seen the show Ted Lasso? Have you heard
Ted
of it?
Lasso, yes, I've heard of the show. I may have seen this someplace.
That's a transition from American football to soccer as well. It's kind of fun.
Oh, I see.
All right, so you've had a little time to settle in. Now I'm going to start bombarding you with questions. Not questions from the audience yet. We'll start with the question I open with every single time, and it's because a lot of students think that we were just like born for this job, right?
Yeah.
But like 6-year-old Francis probably didn't envision he'd be playing FIFA Soccer while talking about virology
Definitely
in Tallahassee, Florida, right?
Yeah.
So there was a winding road. There were many forks in the road. There were events all over the place that, led you to where you are today. So we're starting with 6-year-old Francis. What were those events? How did you narrow down to sciences and then to biology and then to virology and grad school and postdoc, so on and so forth?
So I think many people would have the same story, as in what I did.
Oh, you got to keep playing.
I gotta keep playing.
The challenge.
Yeah. Jeez. Okay, come on, move it, guys. All right, so. All right, there you go.
Nice control.
Too bad. So well, I think when I was a kid, I really liked. So I was the third kid in my family, and third kid in my family, and oh geez, this guy's just running.
I think this ball belongs to him.
Nice.
Third kid in my family, so I didn't have much. What do you call it? Attention. Oh man, what am I doing?
You were the youngest of 3?
Yes, I was. No, we were 4, so I was the middle kid. And so I had all the liberty to try out whatever you want. And what happened was I was,
And they're offside there.
interested in mixing things and stuff. And I remember watching some TV where there was this dog that they gave something to the dog, and
Quite a collision that.
and the dog grew this giant dog, and it was a fun series.
Defenders
And
Curious, do you remember what the series was?
I don't remember. So I remember there was a series called Giant Robot I used to watch. And there was also this other series about this dog which grew very tall.
Oh.
And so I started mixing things.
This was an Indian show?
No, this was an American show. So
Clifford the Big Red Dog?
probably, it was not a red dog. If it was a fluffy, oh geez, man, that was a close one. Oh, it was, okay.
I'm genuinely curious. So it wasn't this one.
Yeah. No, it was not that one.
Okay.
No, it was another one. It was a white dog
That's
with a
amazing.
lot of hair. And that I can tell that, oh, geez.
Oh, man.
And,
That was it. You were like,
yeah, and then I started mixing stuff. I was, oh, geez. What is this guy doing? Oh, man, he's got another goal at me.
In your defense, though, it took him way longer to score this time.
Jeez, man, I was like, you guys showing off.
The replay.
Yeah, like, hey, come on, get out of here. All right.
All right, you learned you could grow dogs.
Yeah.
You decided to try it.
So I started pouring like ink and whatnot I got from my hands. I would just pour into this plant and see if the plant turned blue in color. And I eventually ended up killing it because I kept pouring it every day.
Sounds right.
So I always liked doing that, but I was never good at studies, in a manner of speaking. I was always like to do extracurricular activities, run around, and do things. I did well in my classes, did not do perfectly well. Then there was this math teacher, right? So he would come, and you have to imagine this in an Indian setting. So in India, I went to a public school, and we were 210 students fitting in a room which is barely like twice the size.
Yeah, that's crazy.
So we're all sitting on each other, and all of the this math teacher walks in, and he started teaching, and he was, I think we were doing what do you call it, algebra at the time, okay? And he was teaching. So what he would do is he would come in, he would start a problem from the textbook, right? There'll be like 10 problems from every section, from every topic. He would just start one problem, and then he would, for 1 hour, he would just talk about a story, or he'll just give real life examples of something else. He'll just tell a funny story, or sometimes, right? And then by the end of his story, he would have actually solved that problem. And I was the one person of the 200 people plus, right, who actually got it. Okay?
Yeah.
And when I got it, I was so excited. I was like, this is magic, right?
Yeah.
So I would just go back home and I would practice this, all the other questions in that topic based on what he taught me on the day. And I would do really well.
That's awesome.
Before I knew it, I was writing questions for my math teachers that they could not solve, right? And I would teach the other kids. And so I immediately figured out, I felt like, oh, geez, I'm on top of the world. And you have to imagine, we come from a very poor family, and I've always believed that working hard and working hard and being responsible is the way you can get things done, or achieve whatever you want to achieve. So I always believed that I would do that. Yeah. And then what happened was, then I went to college, which was like an hour travel by bus. And then I realized there are all kinds of people who are, rich people coming in car. I could only go by this public transport system back then.
Yeah.
And, but I was always, when I went to college, I had scholarship to go to engineering school. But then when we went there, they had this list saying, oh, you can do microbiology, biotechnology, and then molecular biology PhD, and then go this route. I told my mom, this is very cheap, right? I'm going to do this. I'm going to be studying for the rest of my life. And she said, okay. And that's when my journey started.
Wow.
Right? So the way I got into learning, okay.
Start another one.
Oh, geez.
So we lost what? 3-0?
3-0. Okay, let me find another team. International.
And Lori suggested that it's this show right here. Wowzer?
No, it was a real animal. It was not a cartoon.
It wasn't a cartoon.
It wasn't a cartoon.
Oh, man. Lori, that's a good try, though. If anyone's curious, here's Wowzer. It's actually a Japanese show. the big white dog. The reason she knows is her dog's name is Hauser, and they ask if she named it after the TV show. There are many big white dogs, apparently. All right,
Let's choose
so,
different jerseys.
Oh, that's good.
Red is good for me. Away, white is good for them.
And you're playing against MLS. Do you watch any of the U.S. Major League Soccer?
No, I just thought the men's team would be bad in the U.S., so I just went for that.
That's the reality.
Okay, let me see. Dida, I know Dida, Cafu, Maldini, I know Gattuso. Yes, Pirlo. Yeah, Pirlo is, if I'm not mistaken, Kaká is there. Yeah, Crespo. Wow. Okay, I know this team.
That's awesome. All right, so part of your journey, you've jumped a lot of places. So what made you decide you went from India to Italy, back to India to the U.S.? Like, what was guiding those choices?
It was very simple. I didn't have money.
Well, that does it.
Yeah. So when after my college days, what I did was, oh, these guys are fast, man. Look at them go. Come on. Yeah. Roundhouse it. Come on.
Does anyone know if they have fights in FIFA Soccer?
Yes, they used to. There are ways to do that, actually. I remember.
So I grew up in hockey country, right?
Oh, right.
Fighting's intrinsic to it. And then you go to a soccer game and you watch people fight, and like they don't throw punches, they throw kicks.
That's right.
They'll like bump chests and kick each other, but no punches are thrown.
Yeah. Run, dude. Come on. That's the way you do it.
Your strategy is injure the other team.
Yeah. So, after I finished my undergraduate and my master's schools in India, right? I actually worked as a lecturer teaching, but I was too young to teach at that time. I realized, come on, guys. Come on, it's USA playing soccer. Let's do it. Oh, jeez. I'll be the one who made USA win in soccer, by the way. Okay. Go for it. Go for it. Yeah. Silachi, where are you?
Oh, nice.
Nice. Go for it. Oh, I went too fast. Okay, I just need to learn how to dribble. Okay, so, and I didn't, I was earning about $100 per month. That was my salary, and I would just give it to my mom, and she would send my applications out, right?
Oh, wow.
I could not afford to take a GRE back then, okay? Whoa, thank
good
God.
stop.
Let's go.
Pirlo.
Andiamo.
So this is like the 4x5 small screen. It's not even a widescreen, so it's hard to tell where you're kicking.
Oh, jeez, man. Thank God. Can't let US win this game against AC Milan. One of my favorite teams. Come on.
It's rewriting all of soccer history right now. All right, your mom sent out applications. This is for a PhD program.
Yeah, this is for PhD programs, right? And I would not, so back then what we would do is there was no internet. And I mean, there was internet and I would write, but most of the applications went on post, right? My mom worked in the post office, so I would just send it through her. And it would cost me basically $100 to ship packages from there to here.
Jeez, that's expensive.
And I couldn't move. I couldn't go to the UK or the US because of, because I could not afford I had to, if I have to leave the country, come on guys, yeah, that's all right. If I had to leave the country, I could not afford the flights and everything. So in the meantime, I was writing like 10 emails a day to professors and stuff. And they would, oh geez, go for it. And I would get some responses, and that would be cool, and I would keep it. And there was one of these professors who actually kept in touch with me. He was studying DNA replication in Italy, and he said, okay, we were talking for a year or so, and he said, if you are, why don't you give this exam for this university, which is a very special school. Okay, believe it or not, it is called the Scuola Normale Superiore. It it is a school where knights were, it's 1,000 years old building where knights were
Yeah.
used to be trained and stuff, and but they had made this, Napoleon Bonaparte in 1810 or something like that. Around that time, might probably be getting it wrong. He actually discovered, this place in Italy, and he named it Scuola Normale Superiore. Okay, like a counterpart for the superior school in France. Okay, which is École Normale Supérieure. And since then, the policy is they take in 40, 50, 500 students at any time. I think the maximum is a small number.
Wow.
That's the whole population, but they pay every student. They pay for everything. They paid
Wow.
my flights, the accommodation, and all of that. And that was awesome, right? And that was the only way I could go.
Yeah.
And it's a European premier school in Europe, and that's how I ended up in Italy.
Yeah.
And then I realized Italians are very creative people and stuff. I was rotating, in a few labs. The rotation back then was just 1 day visit. You interview with them, and that's about it. And there I really realized there was this person who was studying HIV, which was a very familiar topic to me because I had done some work before on that same topic, right? And she was actually labeling particles fluorescently and seeing it.
Oh, wow.
And when I looked at that, I was like, you can see viruses? And that was my expression, right?
Yeah.
That is so awesome. And that's how I got into it.
And that's just, yeah.
Yeah.
That's it. That's what you still do. So postdoc after that. But you went back to India for a while.
Yeah, so it was an 8-year-long PhD because it was not easy to label the virus without killing it. Okay, that was the whole...
You actually bound the molecule to the virus.
to the virus, right? So you're modifying the proteins and stuff, and so you could not kill it. So, trying to figure out what's going on. We just figured out how the labeling works for the ATMs, right? So I was totally, giving up at that point. But my PhD mentor, she told me, hey, you are ready to become a PI now. I said, okay, fine, I'm going to try to come here. And there was this very sweet man. He gave this talk at Cold Spring Harbor. I was watching his video and I said, oh, this is a cool guy. Let me write to him. I wrote to him. By then I had already had offers in HIV labs. Like I had 5 offers. He was the last person I contacted and he was ready to take me on. Okay, I was very frank with him. I said, look, I take a lot of holidays, but I work hard, but this is what I want to do. And I knew what I wanted to do. So when I came to do his postdoc with him, and he hired me for something else, but I started doing something else and I wanted to do that. But he recognized, he said, okay, do it, but if it's failing, don't do it anymore. Right?
That's fair.
And so I started doing it, and because what I wanted to do was start seeing the viruses it takes about 24 hours to complete. 10 hours to complete reverse transcription, 16 hours to complete integration, and 20 hours or so to start seeing the whole infection process. I wanted to visualize the entire thing.
Oof.
Yeah, this was my ambition, right?
Yeah.
But the problem is you have this virus particle that's about 120 nanometers in size. It's got a lot of proteins in it, right?
Yeah.
And you could package only a few hundred molecules of fluorophores inside these particles, yeah? So now you have to shine light on these fluorophores to make them fluorescent, okay? And they can only take so much of light before the fluorescence is completely lost, right? So we have to figure out ways to image them in such a way that you can actually see them for 24 hours while being able to track them inside cells, yeah? That was a challenge, and it was funny enough. We did it, and that's. And then I did a short postdoc, 3.5 years, and I was writing grants at the time from Emory University, right? And the department noticed that I was writing grant and scoring, and they promoted me to a faculty status over there, and eventually, just before I did the interview here, I had a position. It was there, and I said, no, I'm going to FSU because then I got a call over here. That's how I ended up here.
So that was research faculty?
It was a research faculty.
Okay, so you wouldn't have had to teach there.
I didn't have to teach there. It's a medical school as well.
Yeah, but it would have been soft money then.
It is soft money.
Oh,
That's right.
it's, yeah. All right, let's do half 2. Tie game. It's good.
Is it 0-0? I hope they didn't score.
There's only 2 shots on goals, and I think it was 2 penalty kicks.
All right, let's go, dude.
So Lori has a comment. I thought it's strange that 2 to 3 of our international students ask her every year if we provide a travel/flight stipend to get them to Tallahassee.
Yeah, because it's not easy for internationals.
I mean, so it's uncommon in the U.S. to support that,
Yes,
right? Like, but in Europe, apparently, there's at least some programs.
There are some programs. There are very special schools that actually do that. And, you know, I could not have done what I did if I did not have the support. Come on. Whoa,
Got a
man.
shot on goal, ladies and gentlemen.
I still have not crossed the...
Oh, that's fine.
Yeah.
We're making progress.
Yeah, all right. By the end of this game, end of this day, if I score 1 goal, I think that's a success. If I win a game, that is like super success.
So we're 30 minutes in.
Okay.
Flies,
So, yeah, all right.
Now, if you guys have any questions, Ask a Scientist Game and Mediocre Gameplay Expert Science. The questions are facilitated by you guys. So if you want to know anything about virology, host-guest interaction, infections, HIV, antiretrovirals, feel free to ask those questions. Francis is happy to talk about them, as well as fluorescence microscopy.
Go inside the DM.
Maybe a little bit about soccer.
No, dude, come on.
Which I think it's the red button.
This one, yeah?
Kick. Yeah, I think that's.
Kaka, come
It might
on.
be worse. Yeah, just try hitting it when you have possession next and see if it goes long.
I see. Oh, that's right. Okay, got it.
Yeah.
Run.
So that.
Tackle this guy. Oh, man.
That's how we play on the streets of Tallahassee, ladies and gentlemen.
Yeah, that's right. So, go. Whoa. Go. That's right. No, man.
You're on a roll.
All right, let's go for it. Go for it.
Oh, wow.
Oh, man. Come on.
There was something about where you kick
I
it.
know.
Like they were letting
I didn't
I've
know
never
how
seen
to-
that before.
Gattuso, andiamo.
I do envy the European chants during games.
Right.
The U.S. doesn't do like Tallahassee. We do the chop and everything. Speaking of which, have you been to a football game in FSU?
In FSU?
Yeah.
No, I really want to go. Absolutely, I want to go. You know, back then when I was playing football, American football there in Italy, my favorite team was, Philadelphia Eagles.
Oh, wow.
McNabb used to play there back then. He was a quarterback or a linebacker. McNabb, I don't remember. Quarterback. Yeah. I think that's about right. Sounds about right. Yeah. They actually won that year, or they lost it to. I know the team. I was very sad back then when they actually. Come on! Can't let you guys shoot this. Come on!
A free kick given for the tough challenge.
Wait, it doesn't. There's no wall.
No, it's too close.
Oh, okay.
Come on, go for it. Jeez.
Regaining possession in
Andiamo!
I think
Italia!
there's ways when you start getting into this. Somebody can correct me if I'm wrong, but you can start like controlling your team where you give like, if you pause, they might give you an option to do like, you can start like team management. Maybe it lets you set. You can start like being more aggressive or something.
Oh, is that right?
Left joystick.
How do you? Okay. Oh, there you go.
And somebody that knows FIFA better than me.
Substitutions, kick takers.
So go down. Yeah. Okay, that might just be positional stuff.
Settings, maybe.
That'll give you
Difficulty level, game speed. Amateur is great, man. I'm playing expert level, guys.
That's all fine. Let's go.
Match facts, scoring summary.
No, I'm almost positive.
Okay, let's go. Now this is going well. Oh no. How do I do?
Not that one. Nope. Turn it again. One more.
This way?
Okay. Yep. X. Go up to resume match.
Okay, got it.
Yeah. If anyone wants to distract Francis, ask questions now. I'll make him talk about some
No. I gotta be winning, man. So hold on. Nobody talk to me now, please.
8 minutes of gameplay left. Plus stoppage time.
Oh, let's go. Oh, good job.
the shot too easy for the keeper.
Hey, even a tie is progress, though. But I understand you want to win this.
Oh man, I chose United States for a particular reason.
You didn't even have the ball. Okay, I thought you got a penalty there. Okay.
This is the only chance for United States to win AC Milan. I'm just letting you know. All right. Okay.
Man, what would be the score if they actually play?
Oh man, that's.
Like,
I don't think AC Milan will agree to play. I mean, maybe they have to give a lot of money for AC Milan. It's a very rich team in Italy, highly sponsored.
They wouldn't do it just for fun.
Okay, it's fun. I'm not sure. Maybe they would. Who knows? We got Messi over here, right?
That's true.
That's awesome.
Yeah.
Right? That's really awesome. I mean, David Beckham turned things around, right?
Yeah.
When he moved here, nobody knew, I mean, what the hell? But he actually was wanting to build this soccer infrastructure.
Wow, it gives you the option.
Extra time?
End match, extra time, or penalty shootout.
Extra time. All right, let's do it.
Wow, that's fun. I guess it's an extra.
Oh, let's go, guys. You can do it much faster. Let's go. Boom, bala.
Oh, there you go.
Why is he not running so the defenders are running faster.
Speed burst. Let me see if there's a button for speed burst.
Come on. Andiamo. Boomba. Bang him down. That's right. Cry like a baby. I wonder if we can just go kick the referee or something like that.
All right. R1 is.
Oh, man. Let's go.
Try the.
AC Milan.
Try the trigger when you have the ball next.
Okay.
That one.
Oh, I see. Oh, now we
Look
are
at
figuring.
that. We got some moves, ladies and gentlemen.
That's right, guys. Let's go. Boomba.
Good challenge in the air.
Come on.
All right, so I'll dive into high pressure science questions as you're playing a little bit overtime.
Right.
So virology in particular, you study HIV.
That's
HIV
right, yeah.
transmission.
Yeah.
Why the choice of HIV? I mean,
So I don't know, for some weird reason, I've been Maldini. Scafu. Andiamo. Oh, man.
The crowd not in the least bit happy about that.
All right, let's go. Hold on a second. Sorry, man. I'm so bad at this. I have to take a fire either answer that question. I'll do this over here. All right, cool. So the question was why HIV, right?
yeah, why HIV? Of the viruses you could study.
Well, first of all, I think I am very fortunate to be studying HIV because there's a lot of things that we know and there's a whole lot we don't know. Okay, so it allows me to apply, ask out of the box questions and, try to find out new things. What we're really studying is cell biology, right? But why HIV? I don't know. Since when I started my undergraduate research program, I was fortunate enough to work with this person called Soumya Swaminathan, who became the assistant director of WHO during the COVID pandemic at the time, right? And she was studying HIV infections in pediatrics patients, right? And I was there and I would see all these kids and I'm like, they didn't do anything wrong, right? And so I got stuck with that. I mean, there was somebody who was actually studying HIV and using microscopy, and I saw all these colors and then you can see these particles. I was like, boom, we have to do this. So it just became more about the technology development and things we can discover with these viruses rather than understanding, virus pathogenesis and stuff, which is a very difficult topic, and a lot of people are doing great work on that.
Yeah.
Right. And so I started becoming more passionate about how viruses interact with cells and how they go through, fundamentals, how they go through, the nucleus, where they integrate, and how does that impact globally the virus replication and stuff. In the event of doing that, we end up, developing technologies because we are not able to do it. So we have to develop new techniques. And as we develop new techniques, these techniques become useful in screening for drugs and, how drugs work and stuff. We published a bunch of papers on that as well. Yeah.
Oh, we got a raid. Draw Curiosity, thank you very much for the raid. A fellow education-related streamer. You guys should check out Draw Curiosity. Thank you for joining us. How was your stream? How did things go tonight? Looking at the title. Reading the Direwolves paper. You read a paper on stream? That is amazing. that's way better than watching us tie at soccer.
What do you think about the Direwolf story? I'd like to know.
What direwolf story?
So recently this company claimed that they were able to...
reproduce a direwolf.
Yeah. They were able to, re-engineer a direwolf or, but all what they did was, activated a few genes in a
Expressed them.
wolf, of a dire wolf that has gone silent for a long time. Right? There was this whole paper on Nature where they wrote a perspective about it, Science, where they wrote a perspective about it, saying, this is not real, this is not true. It came on Times magazine and stuff.
Well, Draw Curiosity said it is a modified gray wolf.
That's right. Grown in a dog, I think, if I'm not mistaken. Yeah.
De-extinction is not really a good pursuit of
That is
conservation.
correct. That is correct.
No, that's really fun. So you guys look through the paper on that. Yeah. So anyone just joining us, again, thank you for the raid. We are Ask a Scientist Gaming, mediocre gameplay, expert science. Our guest today is Dr. Ashwanth Francis, who's playing a little bit of FIFA Soccer for the first time in about 20 years, while we ask him science questions and ask him to talk about his expertise in virology. And so he studies HIV transmission and propagation and antiretrovirals, and does it with things like fluorescent microscopy and cryo-TEM as well.
Yeah.
Right. And so he wants to image these processes and image infections in real time. And so, we ask him questions while he's doing a whole bunch of playing soccer, which is kind of
Hey,
fun.
look, I got something over here. What's that?
Ooh, that's fun.
Ooh, bala.
Which project or research experience are you most proud of or found most interesting? Let's start with most proud of. I like that one.
Most proud of what we've achieved so far,
Or just which research experience are you most proud of? I mean, this could have been as an undergrad. This could be your own research. This could be anything.
My PhD, I would say.
Yeah.
100%. Yes. I'm most proud of it because it was a complete failure. And I went through it and completed. And because of those failures, I learned so much.
Yeah.
Right? It actually set me up for success.
But you developed the methodologies.
Yes.
Right? Like it's the actual plan didn't work, but the tools.
Yeah. So because,
that's
by
not a failure, right?
well, exactly. But by what do you have to show when it comes? I didn't have many papers. We had a small paper. Maybe nobody studies. I keep telling people to go and look into that because it's got a lot of details on how these things work and they can use it. But
Draw Curiosity. Thank you again for swinging by. Thank you for the raid. We really appreciate it. We'll hope to catch a stream in the future. And every 2 weeks, Ask a Scientist Gaming. Swing by. We will be here with different guests across all disciplines. We've had biology, chemistry, physics. We had a music theorist.
Oh, wow.
We'll have a dance professor on this summer.
Wow.
So a lot of different expertise.
Yeah.
But tonight, virology, if you guys have questions.
Yeah. Hey, so what is this raid?
It's basically people were streaming on a different channel, and then they brought their viewers over.
Oh,
basically.
wow.
Yeah. And so it's like
That's
a social
awesome.
aspect.
Hey, thanks a lot for that. Yeah, absolutely.
Yeah. Negative results are still important and still more common to publish negative results. Scientific Reports is precisely for that. Did you guys publish your methodology?
Yes, we publish it. Hey, how do I? Oh, boombala. There you go. In your face. Okay, come on, dude. Let's go. Da, 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 da.
All right. Middle of penalty kicks. Draw Curiosity has a question.
Oh, no.
What aspect of virology do you research and which virus?
What aspect of virology we research and which virus? Come on, there you go. We study how viruses actually, enter the nucleus of cells. Oh, there you go. I got it. Sorry about that. I was so excited. So what we do is we study how viruses infect cells, and the type of virus we like to study are the so-called retroviruses because they have an RNA genome. The RNA has to go through a DNA, and then this has to go from the cytoplasm into the nucleus, and then that has to become part of the host cell by integrating into the host DNA, and then new RNA is produced, and so they go through the entire process of life. We like studying those viruses. What we want to understand is how did this virus actually know how to get from point A to point B, right? What are the road transports it's using to get there? These are the type of questions we really love to know.
I love the fact that you do this with microscopy because you literally get to see it, which is really cool. Not a lot of people get to do that.
Let's go, Crespo!
Round.
Umbala!
Doing it, ladies and gentlemen. If you guys are interested, here's a link to Francis's group's website. So you can check out his research as well as papers he's published and things like that. Relatively new to FSU. You started last year?
A few years ago. I don't remember. I think it's 21 March. 21
okay.
fall, yeah.
Yeah.
Right after the
Was it
pandemic.
fall? Okay.
Yeah.
And they have to score the next one.
Yeah. Chichi!
Are you winning? Did you just win it?
That's right! I'm so glad I did it, man. We scored 2 goals and we won.
You guys rated at an opportune moment.
That's right.
So the first game, it was 3-0.
That's right.
This game, we took out the win.
I'm doing so much better already.
Pictures in. I took some pictures from some of your papers and put them in a tweet.
That's
actually.
right, yeah.
Yeah, you guys can see. Here's a Twitter photo of. I don't remember which paper this is from.
There are videos. You can. Could you get the videos to play?
Maybe. I don't have my VPN active right now, so I might not be able to see the papers.
I see. No, because there are videos.
Do you have Blue Sky? No, draw Curiosity. I do need to move to Blue Sky.
I am on Blue Sky. Yeah, that's right.
You're on.
Yeah, I had to. I was forced out of Twitter.
Yeah.
Forced out, yeah. Because all of a sudden things started, I was there because I was learning a lot of science and people are posting science topics. People I was following was, doing all kinds of stuff. And all of a sudden it went from that to something unrealistic.
Garbage.
Yeah, it was just things I didn't want to see, and it was troubling me in the same way, right? So, I'm just out of almost all social media. I went on Blue Sky. Looks like a nice place, but truth be told, it's also a little boring in Blue Sky. Not like the original Twitter effect that we have. I think, but it'll pick up. It's very new.
Yeah.
But a lot of the colleagues are there. We are only now starting to see. I'm starting to see scientific publications and stuff. That was pretty cool. Yeah.
Yeah. Draw Curiosity. I plan. That's my summer project after the semester is over. Actually, I can do it now. Now that graduate admissions is over, my life becomes much more flexible. But yeah, the plan is to transition. Ask a
So how many people do you get from the U.S.?
From the U.S.?
Yeah, for grad school.
So FSU requires a 2/3 domestic versus 1/3 international.
Nice.
So that's we typically. Yeah, we try to stick with those numbers.
Nice, yeah.
And that has to do with the college and whether you pay in state or out of state. And so
nice.
they kind of advocate for domestic students. Santi Porus, welcome to the stream. Thank you for joining us. Santi Porus wants to know how the understanding of retroviruses can be used in human medicine.
That's a good question. How do the understanding of retroviruses
help human medicine?
So,
we have predictions, actually.
Yes. So,
What do we want to do?
Did we already ask those questions? Or?
No, not yet.
Okay.
Well, you can pick one. Let's put up a prediction for them that's related to this.
Let's start with, it's very similar to this question, right?
Okay.
Yeah. So, there was one more. I had one more. Hold on.
Might have been one of the ones I...
No. But there was one more. Oh. Okay. Then maybe I can talk about it.
Yeah, go ahead.
Okay.
If you want to talk about it.
So one of the first oncogenes, right? Oncogenes are these DNA that can drive cancer, was originally identified in retroviruses. It was called the Rous sarcoma virus or a chicken cancer virus. I mean, people were very curious about understanding what is killing their chicken in California, and all these guys, they started studying it and they mapped the first oncogene, right? The first oncogene that we know comes from Rous sarcoma virus. It's a retrovirus, right? And then they figured out reverse transcription and all of that stuff, right? So that's one of the biggest. I mean, the entire cancer field walks on the backbone of these oncogenes that people have discovered. All right, let me see. Okay, there you go. So that is one example. The other way to think about is also is Maradona here? No.
2005?
Yeah, no.
No, that would have been too late, right?
Yeah. So the other way to think about it. So we know. So the virus itself, integrates a copy of its DNA into the host genome, meaning it becomes permanent part of the host, right? So that property has been used in the past, and that's this That's this question.
Oh, should we pop that one up?
Yeah, let's pop that one.
All right. So retrovirus gene therapy has been used. It's been attempted at least. All right, I'll post this while you play. All right, so we're going to do a question for you guys related to this. So Ask a Scientist Gaming, we use our predictions for something slightly different. Usually it's used for like predicting video game outcomes, which we could do. Will Francis win this game? No, but we're not going to do that. Instead, I make my guests write questions for you guys. And so you're on Ask a Scientist Gaming honor policy. You can't look it up on ChatGPT or Google or whatever. You just have to know whether you know this or not, or what you've learned from Francis's gameplay, whether you can answer this question. And so it's a question related to the question that was posed to us. So we're going to put up a prediction right now. You guys have 2 minutes. 2 minutes. The question is, what was the first target for retrovirus gene therapy? Was it monkeypox or bubble boy syndrome? The answer is one of those two things. And so retrovirus gene therapy was attempted for one of these two. Is there anything else you want to add to this?
No, that's great. So that's because, they have this property of becoming part of the host. What that means is you can exploit that property to kill
treatment.
No, I don't like this guy. Okay. But you can exploit that property to treat diseases. Oh no, he's coming at me. Get out of here,
Oh no.
Yeah.
But yeah, if you guys haven't done Twitch before, there's a predict button at the top of your chat. Click that predict button and make a choice. Is it monkeypox or bubble boy syndrome? You can gamble as many of these standard internet units as you'd like. And if you're not following us, click the follow button. One, because we like our numbers going up. In fact, we're 2 followers away from breaking 900, which would be awesome to do tonight. But yeah, you get additional standard internet units that you can't use for anything outside of this channel, but you can use them to make us take a drink. Cheers if you'd like that. You can unlock emotes. You can request a factoid from our experts. And so, yeah, make your predictions now. First target for retrovirus gene therapy. Misadventures, welcome back. Thank you for joining us. I guess there's some fun astrophysics news about there about seeing molecules of life in space. Have you seen this?
No. Sorry, man. I'm just trying to win this game because I know the prediction is going to.
That's fine.
Come on.
I
get this pass a little bit.
it was worth a try.
Yeah. Jeez.
But yeah, Misadventures, if you have any news, I saw a popular story. I haven't read the original paper, but yeah, supposedly I don't remember the planet, but yeah, maybe life on other planets. All right, you guys have 5 seconds, get it in. Oh man. So Francis,
that was very good.
Everyone said bubble boy syndrome.
That's right, yeah.
The answer is?
Bubble boy syndrome.
Everyone got it.
Yeah.
So what is bubble boy syndrome?
So bubble boy syndrome is this severe combined immunodeficiency syndrome. You must have seen it in on Seinfeld. There's an episode where he's traveling somewhere and he had to go and see this because he loves Seinfeld, and he had to go and see this bubble. Kids are born without an immune system, right?
Yeah.
So they have to live inside a bubble to protect them from,
It's like HEPA filtered environment. Clean room.
That is correct.
Yeah.
So the problem was, so they thought, okay, they figured out which gene was involved in that, which is required, is mutated and stuff. They want to replenish that. And they used a retrovirus to do it. Yeah. Surprisingly, they cured every single kid they treated.
Oh wow!
Yeah,
I didn't realize it was that successful.
It was. I'm not too sure about the numbers right now, but it was. It was a very big success. But what happened was, come on, Woomba! Oh, come on, go, Jumba! Let's go! Was it a penalty now? So what happened was, in a few years from then, a few kids developed cancer. Okay. Do they know what kind of virus they used in that?
Oh man, does somebody want to put it in chat? Do you actually know? Metaus Bittencourt said the card says moops. That's from the Seinfeld episode, isn't it?
Yeah.
But yeah, if anyone knows the virus that they used, let's throw it in chat. Bonus question, because everyone got that right. Eight people said bubble boy syndrome, which is amazing. Everyone got it right.
Let's go.
Yeah, they made a movie about it as well. I think the boy in the plastic bubble. It was called The Boy in the Bubble, and
is that right?
It's John Travolta's first movie.
Oh, is it right?
Yeah,
wow, I didn't know
that. It was like mid 1970s. I remember this movie very distinctly because I I don't want to spoil it for anyone. But yeah, he has the immunodeficiency, but he falls in love with a girl, and yeah, no, it's fun. They also made a follow-up movie called Bubble Boy, starring Jake Gyllenhaal. That was in early 2000s, I believe.
Oh, is that right?
Which is a comedy about.
I see.
Turns out his mom was lying to him the whole time. Spoilers, everyone. I apologize. So no one has guessed the virus. What was it?
It was a murine leukemia virus, right? The idea was HIV infects humans, so you really cannot use it to treat humans because it causes a disease in humans, right? So they used a virus that is similar to HIV, it's a retrovirus, again, murine leukemia virus. And let's go. And apparently, so what, over the years, people have studied this process. And it comes to true that murine leukemia virus can integrate its DNA close to oncogenes and in promoter regions and stuff. Yeah, and that was the reason why. I mean, it causes leukemia in mice, right?
I see.
And
you want another one?
Thank you. And that is how that's one of the reasons why they don't. So they had to scratch it because there is a basis for you're using a treatment, but the treatment eventually causes the disease. That's not good, right? But they managed to cure, like the kids managed to have an immune system and live for longer.
Yeah,
that was the beauty of it, right? So in proof of principle, it can work, right? But now we have this CRISPR-Cas9 and stuff, so we can carefully do edits. But the problem again there is the efficiency of those edits are going to be very, very small. People are working towards improving that.
So viruses are really good at it.
They're very, really good.
Yeah.
Because they've evolved over, thousands of years. Right?
Yeah. No, that's intriguing. So they cured Bubble Boy syndrome, but gave the kids cancer.
Yeah.
That's kind of brutal, but that added, a decade onto their life or something, right?
I know. They added, some of them lived for much longer than that.
Yeah.
I think, about 20-30%, I think, the ones who developed cancer. Not everybody died of cancer.
Yeah.
But there were a few of them who did develop cancer.
I mean, so could you do that with the HIV virus? Because HIV, it's not cured, but we can manage that, like, at this point, as long as you have the right...
Yeah. But HIV is not known to cause cancer. That's because the integration where they park their genome inside the humans, right?
Yeah.
It's not anywhere close to cancer genes.
Yeah.
Unlike the MLV, which prefers some regions,
Yeah.
And many of these kids who develop cancer, all of them, the integration happened very close in proximity to this one particular cancer gene. There was so it's like science, like, whoa, wow, look at this, right? It's a sad story, but we're learning something from this. We can change this pattern.
So did they know that was going to happen?
They did not know.
Okay. Somebody had to have guessed that was
somebody
possible.
Had to guess this, that they did not know because they're not using the virus to treat the disease. They actually removed everything that the virus has. They just used the properties of the virus to transfer functional gene inside the host.
Yeah.
That's what they do, right? So it's not
crazy.
because of the virus they died, it's because of where the DNA was inserted into the human genome. That's what the kids had developed the cancer. Yeah.
Wow. That's a crazy story. So yeah, they used it to treat Bubble Boy syndrome, which is an interesting name. I'm sure it has a more technical medical name.
It is severe combined immunodeficiency syndrome.
There we go.
Yeah, there you go.
But that's awesome. So yeah, they can use viruses to treat diseases.
Yeah, I would argue, HIV-1 virus could be, a much better tool, but it has got its ethical concerns, right? Because of the diseases it causes in humans. But there are other types of lentiviruses as well, retroviruses and lentiviruses that could be used. People are looking into that, but I think CRISPR has shown a lot of promise, and it's the way to go, and people are doing that at the moment. I still want to see that. Oh, come on, Preacher! Let me pass. I would still want to see the use of lentiviral vectors in genetic therapy. Yeah, especially for but it was a lifeline for those kids, right?
Yeah,
that's another beauty.
So is that FDA approved?
I think it was experimental at that point, and
but even now, is it?
It's no, they would not approve it. FDA would not approve it.
And up the field it goes.
I think they've stopped those type of experiments altogether. Come on.
Now that was lucky.
Let me find them.
Offside.
There's got to be a speed boost in this that you can do.
Jumba. Let's go. Let's go, bro. There you go.
Well, the ball wasn't loose
So
for
if you
long.
hold R1, this bump, the bottom
Bottom,
one,
okay.
I think it'll let you run for a brief sprint.
Oh, I see.
You probably
That's why
get
I've
tired.
been doing this a little bit so they are able to
Yeah,
run.
so hold R1 and see if that gives you a speed
Okay.
burst.
Thank you.
So yeah, it's like they get speed boost, but you don't.
I know. Come on, let's go.
Unfair.
Let's go. Kick that guy in the balls. That's all right. I was not swearing, right?
Oh, you
Yeah.
can swear on this stream.
Yeah, okay.
18 plus.
Okay.
Panic over. The keeper has it in his hands.
We have no standards here.
Let's go.
But yeah, if you guys have questions for Francis, he's happy to answer them. We're talking about
Oh,
viruses,
geez, man.
retroviral treatments, gene therapies, whatever it might be,
And the keeper great
Just
breathes
accuracy on the shot.
with the stop.
Good keeper
Man,
and the keeper.
boy in the bubble.
Yeah, that's yeah.
So how do they treat that now?
No, I don't think there is a treatment yet.
Oh, really?
Yeah, can you
So
just
they're-
look up? Can you look that up? Yeah, I don't think there's a treatment yet.
Wow.
It's a genetic disorder, right? Most genetic disorders don't have treatments.
No doubt about that. It's a goal kick.
It has a treatment, but it'll give you cancer. Boy in the bubble. Oh, there's a 2004? Oh, that's a music video. Never mind.
Aymar.
Bubble boy syndrome.
Yeah. Boom, bomb. Let's go. Let's go, bro.
By the defender.
That guy in the nuts.
Severe combined immunodeficiency.
Yeah, severe combined immunodeficiency.
Story of David Vedder. So that's the one that I think the John Travolta movie's based on.
He met that with precision.
Well, it's
So there's the Here's the disease.
He slid in there and missed the ball. But
is
kept
that
the
treatment
fouls
for
off
it?
the
You can
referee.
look for it.
Prepare yourself for something to happen. This is going to be interesting.
Come on, Maradona.
most common treatment is bone marrow transplant.
Yes, that's the only way, and that is a very painful and very least successful. What is the success rate in that?
Whoa, that's a good question.
Yeah, the success rate is very, very poor. Even bone marrow transplant has been shown to be. Come on, that guy down.
A full
Yeah,
ball challenge that
has been
goes
shown.
well.
You know, people have HIV. There have been a few cases of people who have been cured by from HIV. They all of them had bone marrow transplant. Come on, kick it, Chuba!
Picked up at the back.
It's so much fun. I'm gonna take a bite.
More recently, gene therapy has been attempted. Is that the 1990? Is that what you were
I think
talking
so.
about? Yeah. Yeah, concurrent treatment of ADA injections may impair the success of gene therapy. Yeah. Yeah, that's the developed leukemia.
Yeah.
There are also some non-curative methods. Laminar flow and mechanical barriers. Sure, that's the bubble.
Kim, would you like a onion ring?
Oh, no, I'm good. Thank you. Yeah, that's it. That's crazy. We still
Yeah.
don't have a good...
Yeah.
I mean-
most genetic diseases don't have a cure
Yeah.
yet. Right. Also became interested in viruses because I thought you can cure cancer with viruses. Because the reason being, you know, for example, HIV, right?
Yeah.
It needs a particular receptor to enter cells. So it infects only a few, maybe 3 or 4 types of cells
It's very targeted.
in the whole body, right?
Yeah,
We have
yeah.
millions of types, right?
No, that's a good
It
point.
just, you know, it goes to, so if you can develop a virus that can only target cancer cells, and when viruses infect a cell, eventually they produce so many copies of themselves that they kill the cell they infect, right? So if you can target a virus to enter only cancer cells, for example, then you could theoretically have an oncolytic virus, which is the whole field. I think they have a few oncolytic viruses being developed and FDA approved. I think so, yeah.
I mean, so how does that work? So I mean, it probably recognizes through some kind of surface phenomenon, right? But cancer is a replication mechanism issue. So is there something unique?
That's right. They have cell surface receptors
that are unique.
That are unique to the cancer and stuff. Yeah.
Yeah, I learned that, that I occasionally go to biochemistry talks, but a lot of imaging and things like that.
Right.
Like the way they treat a lot of cancer now is because it constricts blood vessels, right? And oxygen depletion plus size. Like what a crude way to do it. But targeting particular receptors it's such an elegant idea. But not successful yet. All right, so we're still in this game, right? 3 minutes left. You got
No, we could go. We go to the next game. Oh, now there's 3 minutes left. Yeah, that's right.
You should finish this one. But yeah, if you guys have questions for Francis, he's happy to answer them about imaging, virus transmission, host-guest interactions, how virus.
What?
It's a red card. Red card.
Kick this guy to kick the referee too. I'm two people down, by the way.
The pedal red cards. Oh man.
Look at that one, yeah.
It's over.
It's not fair! No!
All right, so we finished FIFA. We could go back to SNES, FIFA Soccer, if you want to keep playing soccer, or we have Super Mario Brothers. We also have a bunch of arcade games.
Let's do something easy where I can play and I'm not fighting with people.
Yeah.
So I can actually talk on the...
You want to do some Mario Brothers?
Yeah, let's do Mario Brothers, yeah.
Okay, let me transition. I'll turn that off. So,
yeah, we should ask this other question to the audience, right?
Which one?
What do they think? Is a virus alive?
Oh, that's a fun question. All right, we're going to pop up a prediction right now. If you guys aren't following, click the follow button, get your standard internet units. While Francis plays some Super Mario Brothers. The original came with the NES. This is one of my favorite games. It's 1985, quality video gameplay.
Wait, let me jump. How do you jump like that?
Yep, and if you hold the other button, the B button, it'll let you run fast.
Okay.
All right, we're going to put up a prediction right now. Prediction in this case is a question. The question for you guys is a virus alive? And by alive, we mean traditional definition in biology of alive. Is that fair to say?
That's right, yes. What is alive? Alive is something that moves.
I mean, this is a important scientific question. How do you define life? But there are generally accepted commonalities of what something is alive or not. So the question is a virus alive? Bet your standard internet units accordingly how confident you are in your answer about whether a virus is alive or not. While Francis plays Mario. How long's it been since you played this one?
Oh, a very long time.
This is one of my favorite games. I still play this daily.
Nice.
I don't know if you saw that TV in the living room, but this is my speedrun default. I love playing this as fast as I can.
Oh, that's so cool.
You know, and people still play this regularly.
I want to start off I have a 4-year-old son, as you may know. Whoa, I want to start him off on Mario.
Oh yeah, no, it's good. It's a good game.
Come on. Oh no, I could jump on this guy, right?
Yep.
And then I could jump on him again and then send him out like that. That's right.
Yeah.
I remember that. Whoa.
Well, you should tell your son to hurry up because they're only 0.3 of a second away from beating the perfect Mario.
Is that right?
Yeah. So the world record was broken about 2 months ago, actually.
Oh, really?
As fast as possible. And so with that record, they're literally 0.3 of a second behind the simulated best run you could ever do in Mario.
Wow, nice.
We can't see the Mario screen. I apologize. I did not sit. There you go. Thank you, Lori. I apologize. They were watching us.
All right. Yeah. See, I'm doing so much better here, guys.
But you got to hear the music, Lori. That's really what's important.
Yes.
I completely forgot to transition. I apologize. Man, we were in a scramble to set up the games because we have like 6 different games across 5 different systems or something ridiculous like that. So yeah. But yeah, you guys, thank you for joining us. Any questions you have for Francis while we're playing a little bit of Super Mario Brothers? I mean, you want a soccer game, though.
Yeah, want a soccer game.
That's
1 out of 3, right?
Yeah, it was. Yeah. It was. Did you score a goal? No, it was a penalty kick.
Yeah.
Yeah.
Well, that counts as goal, right?
Yeah, it does.
Just letting you know. Just letting you know.
Yeah.
Because it's goal. Oh, man.
Bros. Super Mario Bros. Come on. Man. All right. Especially the underground level. Love that music. I should learn this on piano. Really need to. Just for fun.
Boom.
This is taking you back. You remember
Oh
this at all?
yes. Oh, Prince of Persia is something I forgot to tell you.
Oh,
about.
Prince of Persia. That would have been a fun
Yeah.
one.
Okay, let me see.
Yeah, when did you play Prince of Persia? That would have been grad school?
Yeah, no, it was much earlier. Prince of Persia was there. It was an MS-DOS game. If I remember.
Oh, yeah. Like the really early versions
Yeah.
of Prince of Persia, not the...
Yeah.
Yeah. No, that's right. Because I don't think it came out for the Nintendo. There was a version for like DOS. And then it came
That's
out later
right,
for like the
DOS.
Xbox. Yeah. Man. Classic.
So when you do this fast run on Mario, do you also consider how many gold coins you collect and all that?
No.
I've always been about collecting the gold coins.
Yeah. And there's some versions of it to get the high score, right? Or collect every coin or collect every power up. And yeah, there's so many different subcategories. What do you think the fastest somebody's beaten this game in?
I don't know. An hour?
4 minutes and 54 seconds.
No way. Are you serious? Wow. That's awesome.
The warps,
Hey, I could jump up here, right?
earlier you could. I don't think you have the height to do it here.
I see.
But yeah, that's the fastest time anyone's beaten.
Look, that
One up.
mushroom is running.
Oh, by the way, you have infinite lives because we cheat.
Oh,
Yeah, so you don't have to worry about one-ups, but for pride, I mean, obviously. Oh, get it.
I can jump on this guy.
All right, Francis, according to their betting points, not the number of people, 94% said no, 6% said yes. Is a virus alive? What is the answer?
But virus, I would not consider the virus to be alive.
No is
No
the
is the
answer.
answer,
So
yeah.
congratulations, you guys got a 1.07 to 1 payout because you bet heavy on no. Congratulations. So let's talk about the definition
why?
of life.
Yeah. So life. So the ultimate definition is can it reproduce by itself? Man, how do I get that 5,000 points?
You have to do the speed fast run. Yep. So holding that, yeah, you have to like hold that and then use
Okay.
to like jump.
Got it.
So yeah, try it. Hold B with the thumb and then press jump. Yeah.
I see.
So
Got it.
that's how you get speed.
All right. So so viruses, by life, and I mean the only purpose, if you think about it as duplicating, right? It has to be able to self-duplicate. Yeah. So viruses are made of the same macromolecules that make that Whoa! There you go. That make up life. Yeah, like DNA, RNA, proteins. Those kind of thing, lipids, right? But they cannot replicate themselves by themselves. They need to get into, although they have all the machinery, they need to get into the host. They use the host machinery to, make more copies of them to multiply. So that is why they're non-living things outside a host, right? They're just basically protein, nucleic acid material
Yeah.
that can just degrade if they don't enter a host. But if they do enter a host, they can multiply 1,000-fold or million-fold. Yeah.
I mean, so it's broader than just self-replication, right?
Yes,
It has
yes.
to. What's the criteria? Self-replication is one.
Movement and self-replication is, I think, is the ultimate.
That's a big one.
I just died. Did I die?
I mean, but there are examples of molecules that do autocatalysis that self-generate themselves, right? But they wouldn't be considered alive
No, they're not alive yet.
because there's other criteria. And the reason I learned about this briefly is like a Jet Propulsion Laboratory in California. So I was at University of Southern California,
I see.
but JPL would show up to some of our chemistry meetings, and they're like trying to probe for life,
I see.
and they have to define life.
Right,
if you're
right.
looking in space, what is
Right.
it when you have life?
Right.
And it was something like process materials for energy and self-replicate. I don't remember what their
Right.
criteria were.
I see. No, yeah. So life-wise, right? So we've thought about when you think about life, you can think about something that moves. Oh, that means that's plant not alive. It does move a little bit, but not as much.
Yeah.
So moving alone is not enough. But then you think about, okay, can it copy its information, make another copy of itself, right? So I think replication has been
That's the big one.
the big one, the fundamental definition, right? But like you said, if there are enzymes that can just continue replicating itself, so but do they do they have information molecules, right? Enzyme, when you say enzyme.
Oh
they
yeah,
have these,
propagate
right?
information.
Yeah, right.
Yeah, that's another criteria. That's a good point. Cuddle Puppy, welcome back. Always a pleasure. Speaking of virology, how can I get my atrocious lip syncing TikTok to go viral? Are you an expert on that type of virology?
No.
Oh man, don't worry. This video, YouTube, it's going viral.
Oh, jeez, I'm dying here so well.
That move. Man, the lip syncing, the auto tune has gotten so outrageously out of control on TikTok and all these videos using computer generated tune. Oh my god. Have you tried auto tuning, Cuddle Puppy? I guarantee you Dr. Francis has 50 million followers on TikTok.
No way.
That's funny, Cuddle Puppy. TikTok is actually banned on FSU campus.
Is that right?
No.
I didn't know that. Oh.
No, if you go to it, the website's blocked.
Ooh. Whoa, there you go. There you go. Jump on that duck. Kill that duck. Let's go. Let's go by it.
Oh.
Jump.
All right, now you speed at the top.
Okay, come here.
Yep.
And
Look up. Oh, it was so close.
I think I still got 400 here instead of the 100.
I mean, it's close enough.
Yeah.
How can I go viral? Cuddle Puppy, we have no advice for you. We're old.
So
We don't know what the kids do these days
what was the question? Yeah.
about the lip syncing going viral.
Oh man, this is when the volcano
I've
rises.
never been more shocked than learning that Florida, of all places, taking national security risks seriously. Florida State at least is. I don't know about the state of Florida in general, but yeah. No, they cracked down one of the earliest schools to actually ban TikTok. For whatever good that did.
Whoa, man! This is what I love about video games. It just gets you off your seat.
Oh, yeah.
You know.
The body motion involved.
Let's go, guys. Let's go.
Going viral.
I think I can do this. Going viral. Let's go,
We're going viral. Not talking about TikTok, but we talked about movies a little bit, Boy in the Bubble and Bubble Boy and various versions of that. One of the questions we usually ask, and it wasn't on your list, I'm going to ask it anyway. Movie or TV show that gets your discipline right and what gets it wrong? Who captures virus, virology, viral response teams, retrovirals, anything that comes to mind?
So I remember I used to, in Italy, I was teaching this. Man, you know, when I talk about that, I'm dying.
It's okay. Infinite lives.
Okay.
Infinite
So,
lives.
no. Infinite lives. That's right. That's the best part. I just wish I don't get bored of this till I cross.
It's fine. We got plenty of games.
No. So I used to take this class about upcoming new viruses, and so So
You don't
that.
focus. Here's Bowser. So you can go under Bowser, you can go over Bowser.
This guy. Whoa. I can't.
Oh, sniped by
No,
fire.
by the way, I cannot. Can I just jump on him and kill him?
No, you have to go over him or under him
I
and
see.
get to the axe.
Got it. All right, guys.
Cannot kill Bowser.
I'm gonna do this. What are the bets that I will do this in this one?
All right, if you die this time, I'll put
I
up a
won't die. I will
prediction.
go through this. I think I know this guy, Bowser. Talking about. I'll take care of that guy. Whoa. No.
You got this. I'm not even going to be able to do a prediction.
Oh.
We don't do a lot of video games predictions. We should.
Oh
They're
yeah.
kind of fun. Like the
Yeah. I got it.
GTs, take that, Bowser.
There you go.
You didn't even get
That
to
was
do
the most
prediction.
happiest I've been for a very long time. Trust me.
That's the power of video games.
I know.
there. It's so much joy wrapped up in winning.
Oh man, that was so funny. Yeah, that was too cool.
Congratulations, 1-4. We didn't even get to do a prediction.
So we were talking about something. You were saying
Movies, TV shows, get virology, virus transmission.
Yeah. So there was this.
Congratulations on not finding the princess.
Oh, I know. Look at this princess. He's almost a boy princess.
That's Toad.
Yeah, it's Toad. Okay, but our princess is in another castle. Thank you, Mario. Hey, how do I? Is that
You
all?
press start. Yeah, it should go. There we go.
Okay.
Yeah, so you beat 1-4. You have 7 more sets of 4 to go.
Yeah. So, when people were running, when I was, one of the things I did, oh, jeez. Boom, boom. When I was in grad school, because it was taking such a long time, I wanted to know whether I can do it. So I said, maybe I should run a marathon.
Oh, wow.
And if I do manage to run a marathon, boom, bah. Jeez, there you go. Right, then I could get my grad school done, and so I
that's
start
your
this
gauge
marathon.
for resilience.
Yeah. So I start this. Come on, jump.
Oh.
I start this marathon.
Did you train for the marathon, or
No,
you just showed up
I
one
would just
day?
run, and I showed up in to so to say.
Wow,
Oh, jeez.
that's a non-trivial.
Yeah, I would
endeavor.
just so you could drink, and you could wake up tomorrow and do half marathons, not the full marathon, of course. Yeah.
Oh, okay, okay.
So, yeah, pass the okay. Good damage. Get out of here! So, I would go for these marathons, right? And what I used to go for the cheaper one that didn't cost much and all that. And what I realized was they it's $5, €5 or something. I used to pay for it, and they would get me to boom. Geez, I could have jumped over it. Okay. So they would, I could stop every 5 kilometers to, eat and drink and stuff. My friends used to run for timing. I always enjoyed going for that.
Just relax
Every 5 kilometers I'd stop. I don't care about the timing. He would be like 10 minutes faster than me, even though he didn't stop anywhere. But I would have already eaten a lot of, chocolates and whoa. Go, bro. Boomba. Okay, yeah. So that's how I am with Super Mario as well. So as long as I make it towards the end.
Yeah, it doesn't matter. Especially
Doesn't
with
matter.
infinite lives.
Speed. Boomba.
But yeah, movies.
Movies, yeah. So I was
viral
trying to-
infection's common, right? Making
Yeah, so
zombies
there is this movie which I'm trying to remember the name of it. Okay, I think Morgan Freeman is there.
I think it's Outbreak.
Outbreak, that's right. And it's they're talking about Ebola virus over there.
Yeah.
And I thought some of the facts. Please don't ask me what exactly those facts were, but I remember. Oh, jeez, why wasn't he not jumping? I always blame the what's it called?
The controller.
The controller.
Exactly. Internet lag.
Yeah.
Dustin Hoffman, Rene Russo, and Morgan Freeman
Yeah.
in Outbreak.
Yeah. Yeah.
And the tagline is "Try to remain calm." All right. Anyway,
I think that was one of the. Oh, nah.
A button is the right button is the jump one.
Yeah, I'm doing that, but see, okay, this is doing.
Do viruses sense our fear? Thank you, Cuddle Puppy. The answer is no. I'll answer that one for Francis while he focuses on Mario.
All right.
Maybe what's crazy is fear releases certain chemical response in the body, and viruses may be affected by that. I don't know.
Yes, it could be, but we don't know those things. Oh,
I mean, there are some, like the zombie trope is one of the common ones, right? There's a virus that creates zombies. There are zombies in biology, but they're mostly parasitic, if I'm remembering.
yeah,
correctly.
but where did people get this from? Viruses create zombies.
I don't know. It's a good
That's
easy storyline.
that's the whole thing, right? Viruses create zombies. I'm like, even there was this Will Smith movie about that, right?
Yeah, the I Am Legend.
I Am Legend.
Yeah.
I mean, how does a virus change a human being? I mean, is it even practical? Is it
Yeah.
even possible?
I mean,
The virus can infect a cell. The whole
Yeah.
thing the virus cares about is to make new copies of itself. It's not going to make you into a wolf or a dead person who just wake up and do stuff.
Yeah.
But what could do that if I have to predict
Yeah.
of all the organisms what could do that, it could be one of those worms, which is definitely not a virus though.
Yeah, there's a species that infects grasshoppers. There's another one that like infects frogs and grows extra legs. Like there's all sorts of crazy biology.
Whoa,
Ooh, sniped.
that's very cool. Okay. So also I realized we were reading books on Neil. Neil loves spiders, right?
Yeah.
And whoa, there was a big jump, and that got this guy. And there's apparently this one, think it was a spider or a wasp, I don't remember. And it would just feed on the brain or something. It was like, whoa, that is a scary one. I was reading it here like, oh geez, this is not good.
Nature is far scarier than science fiction.
That's right.
Make no mistake.
That's right.
No, I actually I read a book at one point. It was like public perception of science or public perception of chemistry. And one of the things they brought up is that essentially every science fiction whatever, it keys up on whatever the new science is and makes it into a bad guy. Right? And so like when electricity came out first, all of a sudden Frankenstein comes to life. Right? And
I see.
When
That
you
was
have,
very cool.
visiting the African jungle, it's a spider biting someone and giving them power. And then you have like nuclear becomes a scary thing. And you have mutants from nuclear, things like that. And then now more recently it's, genetic.
Yes,
we have, solve the genome and all of
yeah.
a sudden make it a bad guy. And it's a very common trope throughout all of history. Take the thing that's scientifically slightly scary, space travel or whatever it is.
And also the whole concept of the alien movie, right? This big ass alien. Sorry about that.
Don't do it.
This big alien, it just grows out of nowhere, and there is no concept.
Very quickly.
There is very very. There's no concept of time and how long it
or
takes.
mass balance.
A mass, yeah, exactly. I'm like, what,
Exactly.
bro?
Yeah.
That doesn't make sense to me. But jeez, flying duck, get the hell out of there.
But yeah, you're right. The growth rate on
Yeah,
it.
it was like, what?
I love that math because if you need bond formation, right? You need to break bonds and make bonds. There's thermodynamics.
Yeah.
If something grows that quickly, it would be essentially glowing as hot as the sun because
Right.
the energy release for the kinetics necessary to make that happen.
Plus, how many copies or how big is it going to grow? I mean, it's not going to stop.
Or you do the math on the self-replication rate.
Right,
It's like
yeah,
every
yeah.
tenth of a second, 1,000 cells.
Why would it all of a sudden stop, right?
Yeah. Meme scientist, welcome for chat. Thank you for joining us. If you're not following us, click the follow button. You'll be our 899th follower. We're missing 2 from breaking 900. So follow us, but also thank you for the first time chat. Their question to you is an important one. I want you to brace yourself. Waffles or pancakes?
Waffles.
That was not even a debate.
Yeah, no way, man. So somebody asked me, so it was like already 10 years in the US, right? So I'm used to, European culture, the great food and all, the beers, the wines, whatnot, just name it, right?
Yeah.
And then I come to US and, I have all these European friends, so they'd always try to make fun of the US. They asked me, so how are you enjoying it? It's been 10 years. I mean, like, US has so many great things, but on top of all of that is waffles and fried chicken. That is what
Chicken and waffles.
they have, fried chicken and waffles. You can't beat that, dude. Some of my friends also tell me, hey, whoever invented this is awesome.
That's awesome.
Yeah.
So when they visit,
That's right.
chicken and waffles.
Fried chicken and waffles. Oh, man.
Meme scientist, you touched on something near and dear to Francis'
That's
heart.
right.
Cuddle Puppy has a response. I just mix melted butter and syrup and drink it. No point in pretending. It is just a mechanism for sugar delivery. I'll
That's
give you
right.
that.
Yeah. When they serve, because we're used to, chicken, meat, and, fish.
Yeah.
These are salty dishes, okay?
Yeah.
You don't eat it with sweet, and you don't certainly not eat it for breakfast. I mean, sausages and bacon, fantastic for breakfast, right?
Yeah.
When I went to this fried chicken place, because I've been in Atlanta, and I thought Atlanta was the, sweetest people, because New York, I was not very happy with the people wise. And when I went to California, was not like, it's all right. But when I come to Atlanta, and Atlanta was like, the people treated me really well, and they were very kind, and I immediately recognized the Southern hospitality, right?
Yeah.
Then I come to Tallahassee, people were even more nicer. That was fantastic.
Yeah.
Right? But in Atlanta, when I was there, they had this fried chicken and waffles when I went for this breakfast. I was like, this is no breakfast, dude. All right. But I did give it a
But
shot.
you tried it. That's
I
awesome.
did try it, and I was like, what the hell is this? I want more of it.
It changed your life.
It changed my life. That's right. Changed my life.
Now you live in the South.
Yeah, I know. It was the best thing. I think US may have a lot of things to offer, but to top all of them, come on, kill that guy.
Chicken and waffles.
Yeah, chicken. Whoa. No,
So you have to press jump while you're on the...
Whoa.
There's like a timing on it. Maybe it's hold jump. I don't remember.
Boomba.
I don't see springboards a lot in this game because I skip these levels.
That's how you did it in 4 minutes.
Yeah. There's a warp. You can go from 1-2 to 4-1 to 4-2 to 8-1, and then you beat the game.
Jeez,
There you go.
man.
Nice.
Get it. 400.
Cuddle Puppy's critique. You didn't have to drive in Atlanta, did you? Apparently you would have hated it.
No, I was riding a motorcycle in Atlanta. I always lived close to work, and I drove the motorcycle 3 times, 365 days a year.
Oh, wow.
Even when it's frozen, I fell twice. I had an accident because my brakes froze. Hey, how does he... Oh,
Yeah, so tap the button, you swim. So the more you tap it, the higher you'll swim. Yeah, there you go. All right, I'm gonna catch up.
remember this.
Oh no, Cuddle Puppy. That's one thing you don't see in Atlanta. Middle-aged or other older motorcyclists.
That's right.
Oof. So you don't
was
ride a
not
motorcycle
middle-aged.
anymore.
Yeah, I don't drive. I have the motorcycle here. I gave it to my postdoc to use because I didn't have. When we moved here, I rented a house close to campus. So I was using the motorcycle to commute. But then all of a sudden, what happened was we decided to buy a house. So I had to, be moving far away. So we had to buy a second car, and it was not useful to have another motorcycle. So, ooh,
Yeah.
Ghost.
And you have to transport the kid.
Yeah, and you
No car seats for
I know,
motorcycles.
that's right.
Cuddle Puppy has a second question. I skipped it a while ago, but do you remember the 1,000-yard stare when Dr. Deborah Brick's face when Trump was telling people to inject disinfectants?
I know. I was like, what, dude?
Oh my God.
Just to be.
Well, careful. We don't want a visa revoked. So,
That's right. No,
holy
just to be on the. Oh, these two ghosts are coming up. Come on! What, dude? I've not seen this stuff happen. No, just to give some credit to our president, right? So he's not completely wrong. Of course, I think the bleach would kill the virus.
It's true. It's not technically wrong.
He's not technically wrong. He was right about
That
it.
is the glass half full of view
The
of murder.
question was, do you want to live? That should have been the more important question. Of course, that's the way to kill the virus. We kill viruses with bleach all the time.
Oh, man.
Ah, jeez, man, these scary fish.
That's what I love about chemistry papers, too, because they'll do, cell studies, and they'll be like, it killed the cell.
That's right,
The
yeah.
MSO will kill the cell. I
That's
got
right.
a lot of things that will kill the cell. It's just, is it selective? Oh, man.
That's right. So he was not wrong, just to be clear.
Cuddle Puppy, I would love to engage with these questions, but I do not want to get Francis deported. The question was, did you guys see how Trump is 6'3", 220 pounds with 4% body fat?
Yeah, wasn't he more built than Rock the Dwayne Johnston?
So I'm 6'2", 200 pounds with, I don't know, 12% body fat. I'm just going to give you the comparison. Maybe those numbers don't check out, but we are not going to have Francis comment on that at all. Lori's redeemed Take a Drink. So Francis, cheers. She wants us to drink and swim. So
All right.
cheers, Lori. Hopefully have a La La Land in hand.
Yeah, and the IPA, man. IPA has been another blessing from the US.
Yeah, you like it?
Yeah, I loved it. Yeah.
Ironically, an Indian Pale Ale.
Indian Pale Ale, that's right.
Yeah.
And
Do they have a lot of Indian Pale Ale in India?
no, we do not have that.
because it was transported from India,
From India,
right? That's why they
yeah.
because they needed high alcohol
Yeah, the
content.
hops these guys used to add to it. I love my Belgian beers. Belgian beers have always been my favorite. But you know, since I've been introduced to Indian Pale Ale, I've been like, oh wow, it's like spice. You know, once you have spicy food,
Yeah,
can't really go back.
it's an acquired taste.
Yeah, it's an important. Once you like it, because then you're asking for more spice.
Cuddle puppy.
Yeah.
They needed alcohol that tasted so bad that the sailors wouldn't drink it on the
That's right. I think she's right. Cuddle Puppy's right. You know.
Cheers.
Yeah, cheers on that. Okay, guys. What are the predictions that I'm gonna make the flag, the maximum points? So
So you're gonna hold
B.
B, and then you have to jump at the edge.
B and A, right? A is jump.
So you gotta hold B and then hit A.
Okay.
Yep.
Whoa.
Need a
that's
little
pretty-
bit of distance. So try it on the ground. So go back a little bit. So hold B. Yeah, you need a little longer. You need some like run time. There you go. See how we got a lot faster.
Cool.
Believe it or not, that's the thing that limits speed runs is how fast you can get going fast, which is crazy. Yes, I missed. You want to guess who this is? S. Stagal.
Oh, I don't know that person.
So here's S. Stagal's question. I love all at the end.
Okay, yeah.
Pertaining to S. Stag,
Yeah,
obviously. What is your most memorable result from your research? Like, did you ever have a eureka moment?
Oh, eureka moment. Oh, that's you know, when I was doing my PhD, I believed in so there are two proteins. You know, two main proteins in my life. My favorite protein has always been the protein called integrase, which actually integrates the viral DNA into the host genome. Yeah, but the integrase is actually encased inside the shell called capsid, and at that point, we were all trying to figure out how the virus enters the nucleus, and because the virus has to go inside the nucleus to integrate its DNA. And throughout my PhD, 8-year PhD, I believed it was the integrase was doing the job to take the virus inside. And I came for my postdoc, had all these plans up just to prove the rest of the world that they're wrong about the capsid. It's an integrase, right? And I also thought by imaging them in real time inside cells, you would get an understanding. Maybe it's a 50-50 chances both of them work the same way. But then when we had 100% result that it was capsid and capsid alone that was required for entering the nucleus, I was like, whoa, eureka! Oh, geez, man. These fish are biting me on my back. I'm so bad at this. Stagal, please don't judge me on this stuff.
Man. Speaking of which, let's ask another one of these predictions.
Yeah,
I wanted to
let's do
ask
it.
this one in particular because that kind of relates to what you're talking about, right?
Yeah.
All right, so I'll try to give context on this while Francis plays, and you guys can do.
Flying fish.
They're hard.
Fish could fly.
At least some of them
Yeah.
can. Nature's scary, man.
Yeah, I
We talked
know,
about it
especially
earlier.
in Mario Bros.
No, in real life.
And
too.
dragon and what? Oh, jeez, this guy
Brain
won't even
occupancy.
jump.
All right, we're going to put up a prediction right now. If you guys aren't following us, click the follow button, get your standard internet units. We're putting a prediction now. Click the predict button. And the question is, what is the size of the nuclear pore complex channel? And so the context of this is stuff has to get into the nucleus. It does so through the membrane, and there's pores that allow that to happen. And this question is essentially, what is the size of those pores? Is that right?
Yes.
And this has to do with like why viruses do or do not get in through the membrane. And it has to do this with the size of the nuclear pore complex.
Yes.
So the question is, how big is it? Less than 50 nanometers or greater than 50 nanometers?
I'll give you some more context on top of that. You know, over the past 30, 40 years, people have, these flying fish are crazy. People have been trying to understand, you know, what is the size of the molecules that can go inside. Whoa. The nucleus and stuff, and they, you know, what they realized was, what they realized was, you know, molecules that are bigger than 45 kilodalton, you know, they cannot enter the nucleus by diffusion. They need something to bind it and take it inside. This is several years of work, right? And people try to look at the structure, and then they, you know, there was this diffusion barrier. So we'll wait for the answers to, you know, then I will tell you something fascinating about it.
No, that's fair. That's a good place to start.
In the meantime, I'm gonna beat these fish, guys. Fish fry.
Yeah, that was one of the things I really appreciate. So Steve Leonard was on Ask a Scientist Gaming, right? And one of the things he brought up that was profound for me, and I never appreciated, is we have all the components necessary to make life, right? We know how to make DNA, we know how to make the proteins, but his big thing was like the compartmentalization, which he works in, right? The membrane formation and formation. And like migrating things in and out of those membranes, all of that is the foundation of life, and we don't know how to control it. We don't know how to do it. And this question is related to that. So what's the size of the nuclear pore? 5 seconds. Someone put a prediction in there now. All right, Francis, what is the answer?
Whoa, run fast now. I can run fast. I didn't know I could run fast.
No, that's holding B. Believe it or not.
Run fast into the ditch. That's right. All right, what is the prediction?
All right, so most of the points were bet towards less than 50 nanometers. What is the answer?
64 nanometers.
It's greater than 50 nanometers.
Yeah, that's right.
Wow, congratulations. We got a payout too. Lori, you got a good payout on this, 8.5 to 1 on this one. Congratulations.
Wow.
The size was 64 nanometers.
Yeah, so, this has been the debate, right? Because the capsid of the virus is about 60 nanometers. It's a cone-shaped structure. The broad end is 60 to 63 nanometers in diameter, okay? The narrow end is about 30 nanometers. If you look at the nuclear pore, it's, for all we knew, it was about 45 nanometers. So how does this whole thing go inside? So it has to disassemble to get inside the nucleus. Yeah. So this is and what we found, we had a label that bound the capsid, but it'll bind the capsid as long as it remains intact. Even if small changes happen, it'll fall off, is what we've learned so far. And that always fell off at the nuclear pore for the complex to go inside. And it was 100% result, right? So we proposed that capsid is uncoated at the pore, right? But then later on, capsid also has a role inside the nucleus. So, how does it do this job? We always thought, okay, there's, some little bit of capsid is still there to do the job inside the nucleus. But our own research, doing this in another way, we realized we were wrong, that there's a lot of capsid that is carried forward inside the nucleus, but there is something happening to capsid at the pore. Okay, and people have used cryo-electron microscopy to look into the cells, and they realized that because their studies in the past, what they did was they isolated the nuclei and they were looking at the diameter of the nuclear pore. They suggested about 45 nanometers or so, less than 50 nanometers, right? And they said, if now we have these very cool methods. By the way, we got one at FSU now, and we're going to be milling cells, right?
Wow.
So if you have to look inside the cell, you have to actually cut the cells open to make this thin ice sheet, and then look at it. So now they have this new technology.
Yeah.
They could do it inside cells, and they realized the pore complexes are much larger. I think Martin Beck's group from Max Planck, they demonstrate there is energy, in the presence of energy, you can actually have this dilated pore. And what they did was, this is just mind-blowing, right? They used the HIV capsid as a physical object to prove their point. Yeah? They showed that the capsid fits inside the pore as a conical structure. Right? Then you say, oh shit, yes, okay, it's the size is about 64 nanometers.
Yeah,
because you can fit it.
Frame of reference.
Yeah, but still, the mystery is, even if it's 64 nanometers is the size, capsid about 62 nanometers in the broad end, right? It is not just an empty capsid, right? It's bound by it's like a sponge. It binds so many proteins in the cytoplasm as it's traveling. So what happens to all of these things? So the size has actually exploded beyond 64 nanometers, but beyond that, the capsid is able to make it through the nuclear pore complex.
Oh.
And this is what we want to understand. So what is the magic? How does the capsid still remain? Even if it fits, there is no space for it to move. Yeah, it has to flex itself. We had this recent collaborative paper which shows elastic properties are there for capsid. They can squeeze it through. But, all of these studies, eventually what we think about it is, we look at it as an efficient delivery vehicle. Okay, it's a payload, right? You can put a bunch of payload inside it, like genes and DNA, to integrate for gene therapy applications, right?
Yeah.
Into these capsids and send them to where we want to send them. Yeah.
That's
Somebody
awesome.
asked about how we can use the knowledge we learned from HIV for treatment.
That's one of the ways. That's really fun. No, thank you. Congratulations again. Who got the payout on that? Lori and somebody else. I don't remember who the other one is, but so we are at 9:43, and so
what is that? 9:43 is the time?
Yep. So
Wow,
we
man.
go till 11. So we would typically start NARC about a half hour before the end of the stream. So you have till about 10:30. Do you want to
Okay.
keep playing this? Do you want to go to jump to some of the arcade games?
How about we'll see if I can make it through this level
let's do
without
it.
another death.
Let's do it. Believe it or not, most of Mario, if you go fast, it's easier.
All right, if you say so, there you go. Let's go, bro. Boom bop.
Keep going.
Go boom bop. Go fast into the ditch.
Also jump. Go fast and jump. Jump. I should have added jump. So you're expecting, these predictions are always interesting, right? Because
Yeah.
not necessarily everyone got Bubble Boy syndrome on the first one. This one, it's, biased towards, man, it's crazy. But also scales, nobody thinks in nanometers.
Yeah,
Like, nobody casually does, at least, like, I mean, a carbon-carbon bond is 1.5 angstroms,
Yeah.
or 0.15 nanometers. It's kind of hard to think in that scale unless you're a chemist or, you know.
So if the capsid goes there without being broken down, right,
Mm-hmm.
into the nucleus as intact structure, it'll be the largest known structure that we're known to pass through the nuclear pore.
Yeah.
In fact, I mean, that somebody made that point. I was like, whoa, that's another way to look at it. Yeah.
So the key to making this work is you have to hold B while jumping.
Yeah, I'm trying to
It's-
like
it's
this.
got- Yeah. So it's like you just press your thumb down. There you
Like
go.
that.
So you keep the speed while yeah. That was better, man. I was getting there.
That was so funny, man. I'm so crazy.
Cut a Puppy wants to know, Dr. Francis, are you in favor of wearing masks anytime you think you might have a cold, or is staying home better? And so this
Staying
is a
home.
common misconception. So colds, viral or bacterial?
The two types, absolutely, right? So there's viral cold and there's bacterial cold.
Mm-hmm.
Bacteria are, strange, and they're even more difficult to cure and treat. And you're relying on antibiotics and stuff, right? Virals, colds, what they say, right? Whoa, hold on, this flying fish. You know what they say, for a viral infection, right? When you have influenza or something like that, all you
you
need
got
to do.
power.
Whoa!
Get it?
All right. So they say if you have like common cold, if you stay home and take care of yourself and eat like soup and stuff, it'll take you 7 days to get better. Or you can simply go to the doctor and you'll be better within a week. So that's what they say, right?
If it's bacterial, right?
No, if it's viral cold, right? Common cold. But bacteria would take you much longer if you don't go to the doctor. Yeah, so if you have a cold, I would say it's always better
Stay home.
stay home.
Yeah.
There's always a lot of time to work.
That's plenty of time to work. Nights and weekends.
If you're-
Contra, welcome back. Thank you for the tier 1 subscription. 10 months of subscribing. That is crazy. What's fun, Contra, is we just recently celebrated 100 episodes of Ask a
Oh, is that
Scientist
right?
Gaming.
Hey, congratulations!
5 years. We just crossed 5 years of Ask a Scientist Gaming, which is kind of crazy. We don't stream that often, but when we do, mediocre gameplay, expert science. Contra, thank you as always for the subscription. You are paying for our drinking habit tonight. You're paying for Proof La La Land, so we appreciate the support. Speaking of which, if anyone has a Amazon Prime account, you should connect your Amazon Prime to Twitch and give us a free subscription from that. So you can actually give us money for the stream from Amazon Prime. Jeff Bezos writes us a check personally.
Is that right?
No.
Oh man, I'm so gullible when it comes to these things.
But subscriptions is how people make money
Right,
streaming, and
right.
we're not here to make money, but it does help us pay for alcohol. So yeah, if you guys want to subscribe with your Amazon Prime account, it doesn't cost you anything. It's part of your Amazon Prime. So yeah. I'll toast to 100 episodes. Cheers, cheers, Contra. Hopefully you have a drink in hand. Yeah, it's pretty
Cheers,
crazy.
cheers.
100 episodes deep. So yeah.
Man, I hate flying fish. I would never thought I would hate them.
Deuce Aqua, thank you for the sub. Thank you for, again, with Prime. Free from Amazon Prime. It doesn't cost you guys anything. So yeah, we really appreciate it. Thank you for joining us. If you guys have questions, throw them in chat. Francis is happy to answer while he battles the fish. His eternal enemy, the fish. It's not viruses, it's fish.
Jeez, man.
But yeah. A hype train just started in your channel. I don't even know how to use hype trains, but thank you guys for the subs. We really appreciate it. Again, we're just here to play video games and talk science. And then Francis, again, thank you for joining.
Hey, how many? How far I am through this, Mario? I have a long way to go before I finish.
You are on 2-3. And so for every level, there's 4 worlds. So 1, 1, 1, 2, 1, 3, 1, 4.
And how many levels are
You're
there?
On 2-3. There's 8, 4 is the final level.
Geez.
So
okay,
you know you're not even close.
Okay, let me finish. Man, they do all in under 4 minutes. That's crazy.
Yeah. Well, I can show you briefly if you want a preview.
Yes.
Actually do it. Thank you for the follow. Count Chocula 456, thank you for the follow. We are at 899 followers, ladies and gentlemen. One more follower and we cross the 900 mark, which would be amazing. So if you're not following, click that follow button. Misadventures, thank you for the bits. We appreciate it. Thank you for supporting our drinking and gaming habit, because that's what we need to do as adults. That's really why I do the stream. Hang out in video.
Yeah, it's actually fun.
I think you're pretty close to the end of the level.
I want to get this level.
Staggle wants to know.
I love Staggle.
Do you think there will ever be an HIV vaccine, or does it mutate too quickly?
No, HIV vaccines, they are finding it. I mean, in the conventional sense of HIV vaccine, I don't think we are anywhere close to that. But they're coming up with innovative ways to get it done. And I'm really hopeful for the future for HIV vaccine studies. But it's going to be a tough one, because we've known for years now. Before I can die, we've known for years that the antibody responses are not enough to neutralize the virus. So vaccine is going to be a tough one.
It's going to be hard.
Yeah.
So what's the treatment for HIV? I mean, it was huge in the 90s. It was a big problem, but for the most part now it's under control. So how? so
Almost, man. I made it.
Oh, man.
Okay, go for it.
Give you time. IT Scrubs, thank you for the Prime subscription. Again, you guys are supporting our drinking habit. We really appreciate it. The portion we get that Jeff Bezos pays us directly pays for our drinking and video games. So we do definitely appreciate it. So thank you for joining us. And a first-time chat. That's amazing. Thank you for joining. Thank you for the follow. We just crossed 900 followers. It's Scrubs. Thank you very much. It's Scrubs. Thank you very much. We are officially at 900 followers. It's amazing. We think you're...
Whoa! Run faster, bro! I'm there, right?
No. Get the flag, though.
Okay, so...
Speed. Don't jump right away. Give yourself some speed.
What? Jeez, man.
You are so close.
I'm so dumb. 800. At least I got 800 in this world. That's good.
All right. Do you want to see a brief speed run?
Okay, yeah. Let's do it.
Okay. I'm going to start.
All right, Dr. Hansen.
This is my hobby, and so you just sprint as fast as you can. Nothing too fancy. It's faster to go down the tube.
Is that right?
Yeah.
So you come to a different place? Huh?
Yeah. So you come out here towards the end. See if I can do this under pressure.
Oh, but you can... You got 400 too.
I mean, you don't care.
Oh,
Just go fast.
All right. Oh, yeah, World 1-1.
This is the 100th episode, I think. No, this is not the 100th. 100th was 2 weeks ago. Actually, we had a surprise stream 2 weeks ago because I didn't go visit DC for an NSF panel. I was supposed to be in an NSF panel, but they canceled travel.
Oh, yeah.
Yeah, all travel's done. So yeah, Ted Goldberg 2 weeks ago was our 100th guest. And so yeah, this is the 100th and first stream.
Whoa!
Yeah, congratulations. You broke.
You don't want to be 99. You don't want to be 2. 101 sounds a good number, yeah.
Oh, I'm not playing well. We'll skip it. All right, we're gonna go to arcade.
Okay, let's go.
All right, so we're gonna transfer. What do you want to play first? So we have PacMan, we have Street Fighter, we have Mortal Kombat, Mortal Kombat 2.
We've done 2 hours, huh?
Yes.
Oh, wow. Yeah, time flies. It was like
Hour-
10, 15 minutes
Exactly.
for me in my time.
All right, what do you want to do? PacMan, Mortal Kombat, Street Fighter 2,
Let's start with like Mortal Kombat.
Mortal Kombat.
Yeah.
You want 1 or 2?
1 is good. 2 is good. It's fine.
Mortal Kombat 2's it looks a little better. if
Yeah,
you're
let's
okay
do
with
2,
that.
yeah.
All right, so you're going to the arcade joystick.
Mm-hmm.
So this guy right here. So I think it's high punch, low punch, high
Oh
kick,
man,
low
I've
kick,
never used
and
this
then
type of.
block. Yeah,
all
so
right.
this is a true arcade. Like
Wow. They can see these things. They know about all these things, right?
No, you can lift it up if you want to show them the arcade
Whoa.
stick. So your camera's right there.
Oh, oh, look at that. That's a big ass.
We are an adult stream. It is
Yeah, all
perfect.
right.
So I'm going to give you a bunch of quarters.
You said there's going to be. Oh, I have the quarters? Yeah? Really?
Well, I mean, it's arcade. It's pseudo arcade.
So, whoa. You know who I love in this? I used to like this guy with his 6 arms or something like that.
Goro.
Goro.
You can't play as Goro in this one.
Okay. So
unfortunately.
this is, I know this guy.
Liu Kang.
Liu Kang, yeah.
Yep.
And I know this guy too. This guy is, okay, I'll take this guy.
How's the volume? You guys probably can't hear it at all, can you?
Can we hear the volume? The
You
cool
guys
thing
have to
about
tell me if it's okay.
Chase, right? This guy's name is? Am I? Kung Lao. Whoa. Oh, this guy is bad. Shh. Baraka.
You guys have to tell me how the volume is.
I'm just trying to figure out.
High punch, low punch. It's good. Awesome. Thank you, Lori. Thank you, Scott. All right. Contra wants to know, deep breaths from me here. Dr. Francis, have you any thoughts on B-Champ or terrain theory that you can talk about?
Oh, somebody has to explain to me what they are.
Terrain theory. Are they the same thing? Let me Google what this is. Terrain theory. Germ theory versus terrain theory?
What is terrain theory? Can you?
Terrain theory argues if the body is well balanced, then germs that are a natural part of life and environment cannot be. Sorry, that was the AI preview. What is terrain theory in a nutshell? All right. Terrain theory, on the other hand, is about supporting your body and immune system to be ready to combat diseases as possible. As a result, there are many therapies that can work with natural and your body to accomplish rather than introducing something foreign. So the idea is like train yourself.
Yes. And the other one is?
Germ theory, just standard germ theory.
Huh. So what is the question?
Have you any thoughts on B-Champ or terrain theory that you can talk about? And so, like, in terms of preparing your body to deal with things, I mean, that's what
I think preparing your
vaccines
body.
does,
Yeah,
right?
exactly. Yeah, preparing your body to do things is probably the best way to go.
I'll get you there again.
I definitely don't want to take that guy. Hey, Scorpion. This is Scorpion, right? Is that right?
Yeah, that's Scorpion. Yep,
Okay,
yeah.
let's do it.
And so it's like forward punch or something gives the spike.
Yeah. So tell me which one is forward.
So you do this, and I think it's either this one that's
Okay,
punch,
okay.
something like that. I don't remember exactly. If somebody knows Mortal Kombat commands, Mortal Kombat.
I'm not able to do these guys are so fast.
It's
Probably
arcade.
you put me into this very hard.
The quarter eater. B-Champ was a contemporary of Pasteur, Louis Pasteur. So yeah, unfortunately, Francis is not familiar with that. But I mean, there's a lot of thought to be said about like a balanced body can fight off diseases better than an imbalanced to some degree.
Oh, jeez, I'm dead, man.
So the two you care about: high punch, low punch, high kick, low kick.
Okay.
And those are block.
High click, low kick. Okay.
Yeah. So I would try with those first.
All right, let's do it. Thanks.
All right,
Ken.
I'm catching up on things. It's fine. Volume's good. Awesome. Choo choo. We got a Hype train level 2. We don't do hype trains on the stream, but we appreciate your guys' support. So thank you very much. And we are mashing buttons here, make no mistake.
That's right.
Francis has no idea what he's doing.
Oh no, I got this guy.
That's fine. It'll work out. Lori has requested a factoid. Do you want to drop one of your knowledge bombs on them?
Okay. Well, how do I pause this guy?
You don't. Just keep fighting.
I'm dying. This scorpion is.
Oh, Sporty Stats has put the moves in chat. So if you want to do the spear, it's BB.
Oh, look at this guy. He already did the spear on me and killed me.
So the spear is back, low punch. So if you do back, A. You're on Liu Kang now, so that doesn't help you, but that's awesome. Thank you, Sporty Stats. There's the skills right there.
Okay,
so no, let's do Scorpion. Go to Scorpion, and we're gonna do back, low punch, and we're gonna get a spear.
All right, hit that guy.
Right. So I think low punch is this one.
Okay.
So back, A. That's awesome, Sporty.
Oh, before I could do it, he did it. Before I could do it.
Oh, we gotta move. Thank you, Sporty. That's amazing. All right.
He already finished me before even I could start. This is
the arcade version, though. I don't remember if you asked for the arcade. There you go.
No, but I got him and he also got me.
All right. Contra wants to.
Let's win one game.
Can you guys hear the button clicking? I'm curious. I don't know how much sound cancel like directionality because we're mashing away here.
All right,
the camera's shaking.
It's so bad. I've never played on the arcade, you know? I've always played on the
Which one? Super
Nintendo or something like that.
Super Nintendo?
Yeah.
We can do it on this one if I can find it.
Oh, is it right? No, but we can do another one.
All right. Contra has a question. Let's put this one. Are you aware or have you seen the pelomorphic somatids that can supposedly be seen in live biological samples using dark field DIC microscopes?
Pelomorphic somatids, yeah. I think, yeah.
What is that?
It's this type of granule kind of appearance inside cells, I think. Just if you go on the images.
Images?
Yeah, you see it. There you go.
So this is just optical microscopy?
Yeah, just optical microscopy.
That's fun.
I cannot tell more about it.
Well, there you go. There's a link for you guys if I searched images there. I mean, that looks like just normal microscopy, right?
Yeah.
There's nothing fancy about that.
Yeah, look at that.
Huh.
It says these dots. I wouldn't even
what are these structures? What are they for?
I have no clue. All right, let's see if I can get Liu Kang. Oh, let me get this guy.
Baraka.
Shao Kahn wins. Right? That's
Oh, interesting. Contra said smaller than the wavelength of light, supposedly, looking at 150 to 300 angstroms. You can do it with UV light if it's just right.
Yeah.
I mean, but microscopy, we can get smaller than diffraction limit.
Yes.
Yeah. So this
with
is
certain microscopy you can do it.
This is a fun discussion point, though. Is this diffraction limit?
Yeah.
So can you talk about wavelength of light versus size of thing you can observe? Because I don't think people appreciate it. It's only so small you can view with optical methods.
Yeah. Just 1 second. Let me get killed on this one and then I'll talk to you guys, okay? This guy is icing the hell out of me. And I haven't even landed a punch on it. Oh, there you go. I got it a couple clips. Rated. So, so we were talking about optical microscopy and diffraction limits, right? Mm-hmm. So diffraction limit is, the visible length of light, you can start from, probably 400 nanometers to about 700 nanometers. 600, 700 nanometers is what our eyes can perceive and, detect, right? So when you start looking at it in the optical perspective, the nanometers of light is the dimension of light. Mm-hmm. It's 700 nanometers, right? It can shine. If you shine a 700 nanometer light, you're actually exposing an area of 700 nanometers, and you're collecting emission. So the diffraction limit is basically defined by the wavelength, the size of the wavelength of light, right? Yeah. So, but if you think about resolution, right? Resolution is the closest distance that you can resolve 2 points as 2 points and not 1 point, right? And so if you use any of the diffraction limited methods like light, using light, 4 or 5 nanometer light can give you about 4 or 5 nanometers or half that number,
Half that,
right?
yeah.
Half that.
What's that rule? I can't remember.
So it's about 2. It's
not Bragg. It's
no, it's
it has a name.
I know the name. It's just Raleigh's limit.
Nyquist.
Nyquist,
Scott has
Nyquist,
it.
Nyquist.
Nyquist limit. There we go.
Nyquist.
I should know that.
Yeah. So it's half the wavelength of light, right? So so the smallest you can go is about 200, 250 nanometers. But then there's also other optical properties between wide field, types of microscopes are there. Wide field, you get a lot of out of focus light that cannot resolve the 200 nanometer resolution that you can actually get, right? And then, man, this lady is fighting so hard.
You want to go again?
Right. But then you can if you cut out the out of focus light, then you could you could improve the resolution by about 200 nanometers more. Right? So that's the whole idea of this thing. Yeah.
And fluorescence, you can do
fluorescence,
single molecule, single atom image.
That is another, that is kill this guy, Baraka. Kick that guy out. I didn't even land. Whoa. Why? Why is he so much better than me? Yeah.
I mean, arcade, unforgiving. We just crossed 10 o'clock, so we can try also Street Fighter and
Finish it. All right, done.
oh, in the drink, he gave you a fatality.
I know, fatality. This is crazy, man. I didn't know this Mortal Kombat was so difficult.
Yeah.
Yeah, so there are some tricks to it. So you use blinking properties of fluorophores and stuff to allow them to blink so many times till they die off. So you can actually tell exactly where they came from, right? And then you can map these at about 10 nanometers or lower limit. But even now they've come with optical systems, light microscopy that can go all the way up to 1 nanometer.
Wow.
Right? Yeah, that's by using adaptive optics. This is called min field and min flux. Stefan Hell is one of these pioneers. He's a Nobel Prize winner. He got this he invented STED and he introduced super resolution methods, but now he's just really pushing the boundaries of light microscopy for that.
Yeah. So it's no longer diffraction limited.
Yeah, it's no longer diffraction limited. You can go up to 1 nanometer resolution, and you can go. He demonstrated that is possible, right? And you can do this in real time, very fast, temporally as well. That microscope costs about $3 million. So we can't, we have to really work hard to get that, to start using it. But I think using it itself it's not yet mainstream because a lot of people have bought it and they are working towards understanding how it works and how to get that. It is possible to get that resolution from that.
But it's hard.
But you go. I mean, it's if you really like it and just keep working at it, you'll get it. You'll figure out how to use it, and then you start using it.
All right, before I do this follow-up, do you want to keep playing this? Do you want to try something else?
Let's play something else. This arcade is too difficult.
Well, we can go to... We can try...
Stegal, thanks for Nyquist.
I should have known that. I taught this class.
I know. You teach a class.
Thank you, Scott. We're going to assume Scott Googled it. He didn't know it.
Well, he's always about Nyquist. He's always talking Nyquist, yeah.
Well, that's why cryo EM is good, right? Like Nyquist fails at some point. So I get it. All right, we're going to transition. We're going to go to Street Fighter. So this is Street Fighter.
On arcade too, right?
Arcade, yep. So we'll do...
So is there an easy version?
I don't think so. I think arcade is just
merciless.
All right. That is too loud. I apologize. Anyone that has headphones on, I completely wrecked your ears.
Oh, these guys are so good.
Some of these games just go...
Oh, this is...
All right. Contra said...
Oh, I got it. He does a rotating play.
Yeah, unfortunately, I'm switching. I'm using a MAME emulator, and it transitions between games, and the volume has no regulation whatsoever. So I apologize for blowing you guys out of the water. That was very unfortunate. I take full responsibility. All right, so Contra wants to know, Royal Rife and Gaston Nassenens supposedly.
Sorry guys, I'm getting knocked out here.
So you're.
They got it, come on, I'm almost there. Jeez.
All right, so Royal Rife and Gaston Nassen supposedly achieved miraculous microscopes because they were such excellent machinists. There are videos of both, but the Rife 1939 and Nassen's is more recent.
Wow.
The thing Nassen's claims is that the somatid changed shape and function and can have up to 16 stage disease.
Wow.
levels.
Yeah,
And
wow.
So they're using optical.
I'm learning a lot over here.
I mean, I'm curious.
Yeah. Hey, I didn't even start. This guy killed me off.
Yeah, we were talking.
Jeez, man.
Oof. Sorry for waking your dog, Lori. I apologize. I can't tell ahead of time what sound volume is going to be. But I'm definitely looking for more thoughts because so few people have good microscopes to take good looks at somatids.
Nice.
Research is annoyingly scant, but it seems to check out. I mean, that's the hard part about this technology, right? 'Cause
Right.
If only one person has the microscope, there's no
Right.
peer review, there's
Right.
no double check.
Right.
So, I mean, is this something you've heard about? Like these advanced
The
microscopes?
dark field imaging has been around for some time, but they've actually brought in a lot of new, applications for it, and they're trying to mainstream this stuff. But what I am excited about microscopy is Raman microscopy, and
Yeah.
that works in the longer wavelength, and which is got this Dalsin, which I would like to see how they use it, but at the oh geez, this guy, look at this guy's hands, it's huge, right? Whoa, okay.
The long punch.
I'm waiting for come on, I just need to get him on a one round out. Look at his long kick as well, huh? Yeah, the Raman microscopy is something that I'm looking forward to, but they've shown some application where you can actually look at it works based on the vibration states of molecule. Oh, come on.
Yeah, and you can get high resolution.
At a very high resolution, and you can do this without photo bleaching and stuff. Those are things that's why look at this guys just.
Oh, I suppose you don't care about the wavelength. 'Cause it's scattered,
yes.
That makes sense.
Yeah. Oh.
Contrast follow-up. The ability for live blood and live sample analysis, I think, will be a game changer. No more killing tissue to put it on a slide.
That's right. But I wouldn't know how they would even start to go about that, right? The live blood, you have to take the blood out of patients to do that, right? But I think they can find a lot of people doing some really innovative stuff in this. He just burnt the hell out of me, man.
Yeah.
I decided arcade is not for me. it's just been so
But
bad.
that's how they made money, right?
Yeah.
Make it easy. There's no infinite lives.
But that's good, right? Like, so the real players actually match it.
Yeah. No,
and-
people get good.
Yeah, they were good, really good. Okay, Yokohama, El Honda.
We should put another prediction up there. Which one do you want to do? This one's kind of fun.
Yeah.
I don't know if fun is.
Yeah, it's because I think we spoke about it a little bit.
Yeah, but we'll see who's still around that might have.
No, you were asking me something about this guy.
Oh, you want to do that one? Okay, let's do that one.
Yes, that's a fun one. Actually, I have some fun facts about that. Whoa.
I did not edit this one. It's too long. Give me a second. I got to put a prediction up there, but I have to shorten the link because it only gives me 42 characters for these questions.
How do I jump from this guy?
All right, I'm going to put a prediction up there right now. All right, I'm going to put a prediction up there right now. The question is, can a drug stop HIV replication before it infects the cell? And so after it infects the cell is a different story. This question is related to before it infects the cell. Can it stop replication? I only put 30 seconds. I apologize. You guys have 15 seconds left. Can a drug stop HIV replication before infecting the cell? Is there anything I need to add to that?
No, that's good.
That's straightforward.
That was very good question.
Wow, 30 seconds and everyone is leaning on yes. Francis, what is the answer?
Oh, yes, what?
One said yes. What is the real answer?
Yes.
It is yes. All right.
Okay, so we'll talk about this in After I Die out of this guy's fire. Get out of here, man. Hold on.
He's bigger. Should be better.
Come on, corner guy. This guy's got long arms, huh? All right, I died. All right, so that's the new thing, right? So what they did was, so again, somebody asked in the past, as we were talking about the work, why study HIV? Because it is probably one of the well-studied viruses, okay? And we know a lot about it, and we also do not know a lot about it. And based out of HIV, we've actually found so many things that we found through HIV. It's like, how technology developed just by, trying to go to the moon, right? The moon, the entire thing about going to the moon enables so many different technologies, right? Including the plastic bags, small things like that we use every day because of the things that they thought about, right? So it's the same thing when you think about HIV and the types of drugs, the several different ways we try to combat it, right? And that has taught us so much over time. And what we've done is, so most of the drugs we initially developed for the virus, okay, the drugs that we use are called antiretrovirals because it's HIV is a retroviral. Die, man. How do I jump? Yeah! I beat that guy. That guy had like that. Come on, let's do this again. Let's do this. Fatality. Kill that guy.
Wrong game.
Come on now. You can use your hands and catch that guy. What the hell that guy?
After this round.
I'm sorry if I break this. Yeah, geez, this is not
No, you're still on.
almost got that guy. And I won one game, dude. That was awesome.
All right. So the question is, can a drug stop?
How do I pause this? Can I pause this?
Don't think.
Oh, guys, just let me try to survive this one, please.
We'll
I'm gonna give you the answer. There is a drug that does the job. He also published the drug. That was awesome, yeah. So it was called the Molecule of the Year 2024. You can look that up. And not Molecule of the Year. I think it's the Breakthrough of the Year. Breakthrough of the Year. Come on. Kill that bald guy. Yeah, I died.
The year by Science Magazine?
Yes.
There it is.
All right. There are several reasons why it's called the Breakthrough of the Year. And so, so antiretroviral therapy, initially it started off by, you start thinking, okay, what does the virus need to make new virus copies, right? So it needs a reverse transcriptase enzyme that makes its RNA to DNA, right? So that's one of the first drugs they actually developed. And but there are polymerases like that in our body, right? Mm-hmm. So there's a huge side effect that we have. Although you can control the virus, you're also having side effects, right? Then they developed these drugs against integrase, which integrates the DNA into, and they have protease inhibitors, several different. So initially they were like combination of, I don't know how many pills. You had to take so many pills in the 1990s, to survive the virus or to control the virus, right? And somewhere in the early 2000s, that regimen from so many pills became a 3-drug regimen that, was 1 pill per day regimen. That was eventually 1 pill per day regimen. And most people do 1 pill per day regimen. Still, it's 1 pill per day. Yeah. And all of these things target enzymes and proteins that are important for, different steps of the virus. Yeah. But what people have been studying these capsids, and what they've been realizing is that the capsid is a structural element. Okay, it needs to form the structure, and the structure is its function because it has to. It's like you can think about it as a shell that contains a nuclear weapon inside, right? So the virus injects its payload, and this weapon goes exactly where it has to go and does its job, right? But the weapon's casing has to be intact. Yeah, if you mess with the casing, the bomb will diffuse much earlier before it can do the job. Okay, that's how you want to think about this. So people start thinking about the capsid and developing drugs and everything. And Gilead actually came up with this wonderful new drug called lenacapavir. Yeah. So they had this drug for a long time, and what happened was you cannot take it as a pill because it's not absorbed very well in your gut. Then they give it as an injection, and it works. So it's a picomolar binder to capsid. Yeah, and it blocks capsid interactions, and it blocks capsid from delivering the viral DNA into the nucleus of cells. That's what it does, right? By changing capsid properties and stuff. And it's a picomolar binder. It's got a long biological life, meaning because it's got all these heavy arms around it that that it doesn't get degraded very fast in the body, so it can be there at such low concentrations for long periods of time. Now it's clinically approved. Right, people give it.
So what is that, once a month or
once in 6 months?
6 months.
Yeah, well, it's better than the COVID vaccine if you think about it, right?
That's incredible.
Yeah. So what they did, so this, in 2020, we worked on this a little bit as well using the techniques that we developed, what they did was, so they said, oh, cool, this is working. Can we use this as a pre-exposure prophylaxis? Okay, so they just told people to take this drug and just live their life normally. And they asked how many of these people, in Africa, in a clinical setting where there are a lot of people, get new HIV infections. Yeah. They gave these injections to these guys. Go live your life. We'll see what happens. I think 0 people got the virus.
There's no side effects of it.
There is no side because it's a structure targeting drug. How many structures are going to be a conical capsid structure? Only that one virus or 2 virus, that you see, right?
That's crazy.
It's a structure targeting virus. Every drug targets structures, of course, but this is the assembly of capsid structure, the way the assembly works, right? It's long-lived, and resistance profile has been very, very poor, and some viruses they're trying to become resistant, but their fitness is gone completely, right? So you can have drugs. So again, this is a good example of how, studying HIV, we have actually understood we can make drugs that can prevent new infections of, if this is our understanding, and if you can think about, targeting other other types of capsids of other viruses, for example, that could be done, right? Then we could basically give drugs, say, hey, take a pill, or chew this chewing gum, or and stuff.
It's very different mechanism than like a vaccine, right? Like a vaccine uses your own machinery. This is attacking the structure specifically. So contrast, what is its mechanism of action? What does it inhibit? What does it do to prevent injection? Before that, you want to switch to another game?
Yes, let's do it.
Do you want to try the first FIFA Soccer game?
Let's do it. That was very cool compared to everything else we played after.
Yeah.
Hey, how about PacMan? We didn't do the PacMan.
You want to do PacMan? We can do that.
Yeah.
Uh-oh. I put Soccer's up right now. I can close that guy. Give me a second. It's running in the background. Close that.
So the mechanism is the capsid, being I think what I said earlier was it's like this sheet, this vessel that's carrying the atomic weapon inside, right? The vessel has to be intact, and it has to go to the right place, and it has to open up in the right place so that the bomb can be delivered to where you want to deliver it, right? So if you want to think of it that way, the capsid works exactly like that. The capsid has to maintain its integrity. It has to open at the right time for the job to be done. Yes. So this drug, it binds these key interfaces on capsid that are used by host factors, right? And blocks host factor interactions, but also changes that integrity. So it makes an iron shell, right? Either it makes it even more stronger, rigid, or it makes it even more softer. These are the things that it does, yeah? By doing so, it blocks the capsid from entering the nucleus. It just kills the virus in the cytoplasm itself.
That's awesome.
Yeah. It also has effects on the late stages when the virus is coming out. If there's already infected cell, it'll act on the late stages on capsids and block. We are trying to understand how that secondary block is happening as well, but it blocks in multiple places.
That's awesome.
Yeah.
All right, PacMan. We're actually doing Mrs. PacMan. I apologize.
Oh, I've never played Mrs. PacMan, so
Game
okay.
with a bow.
Well, if I lose it,
It's got a bow and lipstick.
okay, this is good.
But
I think I
arcade.
can do this stuff.
Count Chocula, I believe it hyper-stabilizes the capsid.
That's right.
Which is what you're describing. Count Chocula is an outstanding name. Underappreciated cereal from the 1990s. I guess that would have been 80s and 90s. Do you want another drink or are you good? We're 22.
Well, I could have another drink.
All right. We are closing out the 6-pack of La La Land.
Last night. Oh, jeez, man, these guys. I'm just waiting for you guys to come close to me, guys. Come on, I want to eat you up. Whoa! Come on, let me bite your. Come on.
It's really cool if all it is targeting the exogenous capsid.
Yeah.
Which sounds like it is. So
Yeah.
it's selective. There's no side effects. It doesn't end up making something weird in the body. And this is recent. This is
2020 onwards. We've known about this. We've been working.
so how does this change in the future in the next 5
Oh, it changes everything. So that's the way to go. I feel like, it's easier to make, drugs that have these type of effects, strong effects on viruses or bacteria or other processes, right? But we have to target structures. Yeah, we have to target structural elements like capsids. Yeah, capsid's role is to serve as a vessel for transporting the genome and doing, and delivering it to where it has to go. And if you identify those kinds of weaklings for other pathogens, we can design these type of drugs.
So speaking of what Contra said, it might have use as an anti-parasitic.
Well, that would be great because anti-parasitic drugs are very few, right? It's
Sorry.
that would be great to have drugs that can work against worms and whatnot. It's got a very weird sound also, this PacMan, by the way. PacWoman.
Mrs. PacMan.
Mrs. PacMan.
Or Miss PacMan. I apologize. But yeah, if you guys have questions, we are, 2.5 hours into Ask a Scientist Gaming, mediocre gameplay, expert science. Our guest today is Ashwanth Francis, goes by Francis, has expertise in virology, host-guest interactions, HIV, monitoring those transmission events via fluorescence microscopy.
Hey.
And so if you guys have any questions for Francis, he is happy to answer.
Hey, I thought eating cherries would give you superpowers.
No, it only gives you points on this one. It's the big circles give you, yeah, there you go. Turns them into ghosts that can be, biologically accurate.
Right. Come on, Terry. Come on. Oh, jeez.
One of my favorite parts about Ask a Scientist Gaming is it gets increasingly harder through the night as I relay.
I wonder why. Yeah, right.
Because I have many different experts. So last week it was Ted Kohlberg who does high energy particle physics at
Nice.
CERN, right? And I'm not an expert in any of the stuff you guys do, but I'm like trying to relay it as quickly as possible. It's fun. But cheers, everyone. Thank you for joining us. Mediocre gameplay, expert science. If you have questions for Francis about viruses, gene therapy, antiviral, antiretrovirus, throw them in chat and we will answer them over the next 34 minutes. It's going to be time to switch to NARC. Are you ready for this?
Yes, let's do it.
Let me get the game up and going. Do you want to see the timer or not see the timer?
What is that timer?
So I have a clock here.
Okay.
It's going to keep track of how fast you beat this game.
Oh, is it right?
Yep.
Looking at what I'm doing so far, I don't think I'm doing a good job, guys.
That's fine. It'll be good. I've split. Okay, we got a clock up in the corner. Make that guy a little smaller. There we go. Do you want to see the clock or do you not want to see the clock?
I'm totally fine, yeah.
All right, let me transfer this. Sorry, we're changing between computers. All right. So that guy, NARC, switch over. Lori, this time I'm going to make sure you guys can actually see the game. All right, Francis, as is tradition on our stream, we have every single guest play this game called NARC. It is a bargain bin game for the Nintendo Entertainment System. As you can see, just say no, you're going to win the war on drugs. So it's important I give you context into what you're playing because you've never played this before. In fact, Francis, we started a little bit late, so Francis had no chance to practice at all. So he's seeing this blind for the first time ever right
Every
now.
game I played, I think for the first time. That's right.
So this is NARC. No one had the guts until now. You are Max Force. Your mission: bust Mr. Big and destroy his dreaded criminal empire, seize all contraband, stolen money, illegal weapons, use rocket bombs, high-powered machine guns, apprehend all suspects, protect the innocent, punish the guilty, stop at nothing.
Wow, that's a lot of responsibility.
No, you're going to win the war on drugs.
Can you use this?
No, it's the original NES controller.
Okay.
So you're going to have this guy. Right. So I wish you guys could see the setup. We have like 8 different controllers in front of Francis.
Okay.
All right, so you're going to press start button. And I'm going to walk you through some of this so you can get the buttons. And if you guys have questions, put them in chat. We're going to distract Francis while he tries to do this.
Who's this crazy guy?
This is Max Force. Oh, the guy in the trench coat? That's a drug dealer from the 80s.
Okay, oh, that's
All
how
right, so
you
your
do it.
goal is walk right. So keep pressing.
Not this guy's? How about killing this guy?
There's no advantage in killing anyone. So I'm
Okay.
going to walk you through the buttons. So there's A button. If you tap A, it's a rocket. So tap it lighter than that, even lighter than that, just like a little tap. He'll shoot even less. Don't even.
But they're
push it hard.
shooting me.
So go up. It doesn't matter. You have infinite lives, infinite bullets, infinite rockets.
I see, okay.
Keep walking right.
Oh, this is like
So
our
hold,
Super Mario.
hold A, and you'll shoot bullets. Tap A. Just a really gentle tap. Lighter than that. Even lighter. Lighter. Don't even push it all the way down. That's a rocket.
Okay.
And so, yeah, if you tap it, you get a rocket. You're going to grab that card right there.
He killed me.
Yeah, they'll kill you, but you have infinite lives. So keep going. And then B button, if you hold B down, it makes you squat.
Oh, that's... Oh, why is the dog biting my bum?
So if you hold both buttons, you can actually shoot him. So hold both buttons down simultaneously. Turn around and shoot the dog. There you go.
He became a
Through the
puppy.
door. He became a puppy. Physics. So if you hold B, it squats. If you tap B, it jumps. So a little gentle tap. There you go. And so, yeah, that's it. That's the controls. They took 4 arcade buttons and put them on 2 Nintendo controller buttons. Go through the door. And that is level 1.
Wow.
But yeah, if you guys want to distract Francis while he's playing a little bit of NARC trying to win the war on drugs, now is the time to do it. Ask any questions you have. One of the questions we haven't asked that we do on regular streams, we ask pretty much every guest this. Unlimited budget, no moral qualms, what research would you do? And so we're talking, I mean, you could infect the entire population with a virus. You could make the best microscope ever. You could measure viruses on the moon. What do you want to do?
Well, I would definitely be studying viruses and virology because it's really a lot of fun. We learn a lot of things about it. And microscopy would be part of it. But I think, we will definitely improve methods to... What I would really love to do is build viruses from bottom up or virus-like particles, right? That's my, because they are great delivery machines, like we've discussed before during this entire period, right?
Yeah.
The great delivery machines, and we could certainly use them for therapeutic purposes, I guess. And try to figure out if you can actually get the virus to infect cancer cells or things of the nature, right? Is that an injection or
That
what?
is a needle.
It's-
Yeah, this is 1980s drug dealing. So you're going to shoot the guy who's wearing black with bullets. Oh, man.
Oh, this is each line. I see. It's each line. There's a person standing there, huh?
Oh, we got a raid from Actual Education. Actual Education, welcome back. Thank you for joining us, raiding with the party. We really appreciate Actual Education. If you guys haven't checked out, nonprofit tutoring, science, math, actually does like office hours on Twitch.
I see.
So,
Oh, wow.
so anyone can swing by and learn, cover. What were you guys covering tonight? This is free tutoring for math and science, but yeah, I guess if anyone shows up, they can ask any questions. But thank you for the raid. We really appreciate it. Thank you for joining us. We are Ask a Scientist Gaming, mediocre gameplay, expert science. I'm Ken Hansen, Associate Professor in the Department of Chemistry and Biochemistry at Florida State University. Joining me today is Ashwanth Francis, who is an expert in virology and HIV and transmission and monitoring that with fluorescence microscopy. So if you guys have questions about viruses, now is your opportunity to get those questions answered. While Francis plays a little bit of NARC, this is a game he's never played before, but
I don't believe in NARC.
it's gonna be good. But yeah, you can hound him and learn from his expertise. Wow. Oh, Lori has redeemed Take a Drink because you're about to get in a car during the video game. This is NARC Drink and Drive, so cheers, Francis.
NARC Drink and Drive.
We'll pause. We're going to take some more time. Cheers, Lori. Thank you for joining us. And everyone else, we really appreciate it. We get to hang out and drink and play.
So tell me, Ken, how do you come to choose Wednesdays?
Wednesdays? Well, it was one of those things we were debating. Like, I don't want to take people's Friday and Saturday night.
Thursdays would be great, right?
Thursdays. The other issue is I used to teach Monday, Wednesday, Friday at 8 a.m. And I was like, Thursday, I don't want to wake up at 8 a.m.
I see.
Because I get thoroughly drunk during this.
But you could always, by yourself, tell them that, you can't teach on Fridays.
Exactly.
Because I have to do this on Thursday, guys.
But I teach the 350-person Gen Chem class.
Oh, geez,
so, yeah, I don't get
that's a lot. Oh,
flexibility.
geez.
Yeah. So, yeah. Oh, man. So you're going to get in the car. So jump, tap B. Gentler. Like, don't press it down. 7 frames is what you have.
Yeah? Okay.
That was it. Maybe get down closer to the car. All right, tap it again. Gentle.
Why is he walking around through the car?
So you're holding it too long. You're squatting, so even gentler than that. Just like pop. There you go. On the bottom, drive right.
Okay, God.
Oh, man.
Oh, man.
All right, we got some distractions. Keep going right. That was perfect. You saved some time. Stagal wants to know.
Yeah.
Please give us your thoughts.
Stagal is awesome.
Give us your thoughts about the role of viruses in the origin of life.
The role of virus in the origin of life, guys. That's an interesting one. Oh, what the hell is this Mortal Kombat guy coming here?
That's Joe Rockhead, and he's also a drug dealer. So keep walking right.
Oh, geez, why?
They'll keep coming. There's no advantage in killing anyone. Just keep walking right.
Oh man, this is great.
You think about rockets though? That's good.
Yeah, I think so. Viruses would be right among the top of them. In the evolutionary process, right? I would think viruses will be up there, the top. Because if you think about it, they are proteins. They are all the macromolecules, right? Hey, how do I jump over this, guy?
So tap B. It's the same way you got in the car. There you go.
That's cool. Yeah. So yeah, I think so. When you think about it, right? I should have put this other question. Can we add a new question for the people?
You want to add a new one? Yeah. What is it?
What is the fraction of our genome that contain virus DNAs? Or fraction of a genome containing virus DNA? 10%.
Give me 2 options.
Less than 10%. More than 10%.
Less than 10%. All right, on the spot, creation of a prediction. Yeah, I like it. You are losing time, buddy.
Oh man! I'm playing for time.
Yes.
But no, this is
speed run. Right? You are versus every one of your colleagues that has come before.
Oh, you're right. Yeah. That's so good. Fucking guys! Look at me walking coolly and shooting all that stuff.
All right. So, fraction of genome with virus DNA, is it greater than 10% or less than 10%? So this is residual viral information.
Yeah, I mean that'll be a good place to
Yeah.
Staggertal's question was very on point.
Yeah. Origin of life. And the question is fraction of genome with virus DNA. Genome is huge. How much of it is from viral information?
So, by the way, our DNA is about 3.3 billion base pairs of nucleic acids. What the hell are these dogs doing?
They're drug dogs.
Drug dogs? They're all on drugs. They become puppies. That's why. Come on. They got fire in their mouth.
We had a question follow up on Estegal is, please give us your thoughts about the role of viruses in making the 2019 musical film Cats.
Oh man.
Oh man.
It was all viruses that I could tell. Did it go virus?
Oh, no. Speaking of cats, it's a parasite, right? What is it called? Toxoplasmosis.
Toxoplasma, yeah, that's the parasite. Yeah.
It's not a virus. So it's probably a. So stay on the bottom.
Okay.
Go up to the top. You'll run into a wall eventually, but that's fine. Jump over the wall. Keep going.
I figured out how to.
What virus caused the creation of a feature-length fever dream? It was Toxoplasmosis. That was clearly the cause.
Toxoplasma.
Yeah.
So scared of these parasites.
Oh, really?
Yeah, parasites. It's so difficult to get them out, man. I mean, like, even treatment is not that much. I mean, worms. To take out worms from your body, I mean, what would you target? If you're targeting any of the worms' biology, right? You're targeting their replication complex, their DNA replication. You're targeting your own proteins, which there are a lot of homologs there, right?
Get in that one. Stay on the bottom when you drive. All right, so Francis, you create a question on the spot. Fraction of genome with virus DNA, what is it? Less than 10% or greater than 10%?
Greater than 10%.
What's the actual estimate?
I think it's about 25% or something like that.
Wow.
Yeah, it's actually, it could be a lot more than that. I have to go all the way up here and then jump. Okay.
Yep. Keep going.
So it's also retroviral elements, right? So there are all these extra DNA in our body that we think plays a structural role in maintaining, in maintaining life. But they all come from these million years of, infection and, reinfection by these so-called HIV type of retroviruses, right? That leave their DNA inside these things. Oh, who's these Chinese guys?
Why do you assume they're Chinese?
I don't know, Japanese guys. I know because they had this Japanese hat, or maybe they're English guys. They're certainly not cowboys from Clint Eastwood movies, right?
That is amazing.
Yeah. But these English guys,
welcome back. It's been a while. Thank you for joining us. We're talking about virology. This is what the war on drugs looks like.
Oh, really?
There.
Kill me, guys. Get out of there, guys.
I'm gonna go right. These guys,
why are these karate guys doing?
I mean, this is the war on drugs. There are no. Listen, guys. So grab that card. Go through the door.
I'm not able to grab the card.
Not too far. Oh, okay. So once you go through the door, you can't press up anymore. It'll bring you through the door again.
I can jump over these guys, right?
Yeah. So go up through the door, but don't press up anymore. Nope.
Oh, I see.
Just press up enough. All right. Contour wants to know, thanks a bunch for fielding all the questions, Dr. Francis. Have you got any favorite pictures from under the microscope of things that we believe to be viruses?
What does it mean? Sorry, what does it mean?
That's a good question. I mean, we've imaged. Well, like TEM, we've imaged viruses, right? We know what those look like. But yours, fluorescent microscopy. Oh, you're going right. Sorry. There's you got out of that room.
What are these clowns? They're killing me with a dagger.
This is war on drugs.
Okay.
Sorry, I didn't coach you through. Keep going right. Don't go through this door, not that door. There's going to be a key.
Oh, Jesus, crash me.
This right there. I apologize. I missed it. Get that safeguard through the door.
This clown is killing me, dude. All right, let's go.
I mean, so your microscopic images, they're essentially bright spots, right?
Yes.
Because you're fluorescently labeling things. But we have imaged viruses. Like, we do know what viruses look like.
Not every virus looks the same. Right? And not every virus looks the same. And HIV especially, like, we study this capsid, and it's got a very peculiar...
Left on this one.
Very peculiar shape. It's conical. It doesn't have any, same symmetry. It's got like hexagons, hexamers and pentamers, and it's a very peculiar shape. We wonder why it has got this peculiar shape and what makes that so special.
So you're gonna shoot all these guys, shoot them with bullets instead of rockets. So hold A. Yep. And then,
That guy just blew up.
yeah, you shot him with a rocket.
Gee,
What did you expect?
who's this guy?
It's a Rambo-looking guy.
Who's this Rambo? Man, this is the most trippiest game I've played so
It's
far.
amazing, isn't
Yeah.
it?
Who's this guy?
This is
Jeez.
literally a bargain bin game. So you're going to
Yeah.
shoot those green guys with bullets, and one of them is going to drop a green card eventually. There it is. Grab that green card through the door. There you go. Touch the... You got to swipe the card.
Oh, really?
The game's real.
Yeah, swiping machines back then. I don't know.
Sure. I mean, this is quality Nintendo gameplay.
Yeah, you know, I was only scared how we going to pass for 3 hours. 3 hours just went by.
Flies by,
Flies
doesn't
by
it?
so fast. Yeah.
I mean, but remember back to childhood when you'd actually focus on games? Like, man, days would go by,
I know,
right?
right.
Like, yeah.
So this guy has unlimited ammo and all that?
Yep.
That's
unlimited
crazy.
Well, no, I have cheat codes in.
I see.
So we're cheating, but that's fine.
So how do these clowns are killing me, man? It is crazy, yeah.
This is the war on drugs. But yeah, if you guys have questions for Francis, did we answer do you have a favorite image? Like, let's say a fluorescent image or a, like, microscopy or a TEM image. Do you have a favorite?
Oh yeah. So you know we are in the business of studying how capsids uncode, right? How do I? Will that give me cash? 24 hour
Okay,
cash?
no, you don't need cash.
Like what? Cash machine.
I need to withdraw.
Yeah. so you know we are in the business of how capsid disassembles and you know how it uncodes release the genome, right? But more recently we've been developing some tools for studying capsid and its contents, and we wanted to prove that it's actually looking like a capsid. And I had no better, more pleasure than seeing this intact conical shaped capsid.
You have this.
I have that. Come on.
What was the image from?
It was from a TEM.
TEM.
Yeah, it was
You saw the actual
Yeah,
shape
it's the
and-
shape of capsid and
That's
stuff.
cool.
And Stagall was helping us do this over time, and we've been trying to do this for some time. And when we saw it, I was like, what? I never thought I would be so happy looking at a conical shaped capsid. I was like, what?
What would I search to find that image?
Well, you can just look for HIV capsid, EM, and then you'll find some images. Over there.
HIV.
Yeah. Boom, boom, boom. Guys.
Is it any of these guys?
Yeah, that's the highest resolution. See, the
This one?
first one is a beautiful picture of viruses getting out of cells. You see that one there?
This one?
Yeah. That's beautiful. So many viruses coming out of cells. You see this conical shaped stuff? That is the capsid.
Huh.
Yes, that is the sheet for the contents inside. It's really beautiful. That's a very beautiful picture. It's not easy to make these pictures, right?
Yeah.
There's a lot of processing.
Going right.
Oh geez, these dogs are eating me, man. Come on,
mean,
get out of
They
here.
weren't gonna make it easy. Oh, Cuddle Puppy. Who is the bad boy of virology? Slick back, black hair, sunglasses indoors, leather jacket.
Oh, bad boy of virology, huh? Let me think about this question.
Careful.
I might just not get another grant if I say his name.
Yeah, exactly. I don't know how they're gonna.
Hey, I think I missed out my card somewhere. Did I
No,
miss?
so this one you have to shoot the guy. So shoot everyone on screen. Just use bullets, and you're gonna have to shoot a guy in a wheelchair with rockets.
That's a really good. Oh, with the rocket, you have to shoot this guy?
Yes.
I did.
Oh man, I'll tell you who the bad boy of photochemistry is. So we had a postdoc in our lab at UNC Chapel Hill. His name was Akitaka Ito. He's currently a professor at Hokkaido University. But he was, imagine an anime character that dresses in
wow.
slick back hair. That's how Akitaka dressed, and it was amazing. Like he was the bad boy of photochemistry, like straight up anime character.
So I want to say there is this guy, I do not know about virology, but retrovirology, I can tell you, there's this guy called, who I really adore and respect, and he's, this guy called Tom Hope from Chicago, and he will be happy that I told that. Yeah. So, because he's always been about the truth, and but he's also been my fierce competitor, and he's also the person who actually told me, nobody's gonna read your goddamn thesis, submit it, and come to the U.S. You'll do so much better over there. Because he was collaborating with us when.
Tap lighter, a little lighter. There you go.
Timing is right.
Gentle. So you're pushing the button too hard. So like the time it takes to push it down is longer than.
Why is the dog biting me, dude?
There you go.
Get out of here, dog.
Just stay in the middle, kill all those guys, rockets everywhere, just keep tapping it. That is how you're going to beat this particular boss. Oh, that was close. So if you see a bullet come out, you pressed it too hard. Cuddle Puppy's fault.
I got it.
All right, that's one. You gotta get him 3 times.
3 times, no way.
Okay, so Cuddle Puppy has a follow-up. This is amazing. Who's the good boy of virology? Barks when the doorbell rings, wags its tail when you look at him, licks your face whenever it's in range. Who's the good boy of virology? Puppy-related reference.
There are a lot of them, actually. All the colleagues, virology colleagues, they're fantastic people.
Cuddle Puppy. This is why we allow you on stream, Cuddle Puppy. Thank you. All right, a real question. Contra, what size are the capsids?
60 nanometer.
You gotta kill guys on screen.
Yeah. Oh, you gotta kill them also?
Yep. All right, and then rocket him. I'm gonna go to the red.
Oh, I got it. Yeah.
All right, one more. Kill everyone on screen. Shoot a rocket. I'm gonna run to the restroom. What size are the capsids?
It's about 60 nanometers on the broad end, 30 nanometers on narrow end, and about 80 nanometers lengthwise. Yeah. And it's a pretty big structure. If you think about it, the nuclear pore looks just like just the size of the capsid. And HIV has to enter the nucleus through nuclear pore complexes. That's the beauty of it. Whoa, I got him. Oh, these dogs just bit the hell out of me. Got him. All right.
I hear you got it?
Yeah.
Nice. All right, next boss. So I'm going to walk you through the start of this level.
Okay.
All right, so we go to the top of the screen. What you're going to have to do is do a jump rocket. So you're going to have to tap B to jump and then A to shoot a rocket. And you have to hit him in the hat. So you're going to want to run.
Right. Okay. Who's this guy?
Go this way. This is Mr. Big.
This is Mr. Big. Why is he hiding me from the back?
So run this way. Hold right. Get away from him. Nope. Get off the down button. B button. Run. Get away from him. Get some distance. And what you're going to do is turn around, tap B, and then in the air, tap A. Yep. So both of them very gentle. Run away. Do it again. And you have to hit him in the hat with a rocket.
On the hat? Why is the hat so important?
That's just what he has
Mr.
to do.
Big
That's the
International.
rules. His hat is his weak point. It's the Achilles heel of Mr. Big and his drug empire. Estegal has a suggestion for Bad Boy. Mark Wainberg. Said something like he would drink a cup of coffee spiked with HIV because he didn't believe HIV caused AIDS. That's some RFK Jr. stuff.
Yeah, I'm glad I never met him. Because I would have given him some HIV to drink.
Can HIV transmitted via digestion? No, it has to be open in bloodstream.
Yeah, it has to be in the bloodstream, and it's because Like I said, it targets a handful of cells.
Cuddle puppy. And I meant bad boy as an informal and adventurous, not evil. Oh,
Wow.
man. That savage is right. Thanks, Cuddle puppy.
Hey, how is this big
Yep,
being
so run away.
keeping me there? I don't know.
So yeah, keep going. Get some distance. Get some distance away. But yeah, tapping B gently, and then A on the way down. All right, turn around. So stop. There you go. Tap B. Tap A. Oh, that was close. So you're pressing the button a little too hard. Run away. Do it again. This is the rate limiting step for everyone. Yeah.
He's like the nuclear board complex. It's just too small, faster.
It's the barrier. All right, stop there. Turn around. And then jump, tap B. There you go. So that a little bit later on the A tap, a little gentler on the A tap. Do it again. Oh, that was close. That was very close. All right, run away. Do it again. If you guys have any questions, here's your last 10 minutes.
Is this BIG, PPO?
Oh,
Big Papa.
run away. Just tap B. So a little gentler on the A tap. Just enough to like touch the button. Yeah, a little too long. Too long. It's better. Yeah, if you see him squat down or you see a bullet, it means you pressed it too hard or too long. All right, last minute questions. If there's anything I missed, is there any questions on the list you wanted me to ask? Oh, what's this is a fun one. What is the Earth is flat or anti-vax equivalent to your field? Is it anti-vax?
Capsid uncoats in the nucleus. That's the yeah. I mean, because you know, there is all these evidence that the genome and everything, you know, becomes accessible in the cytoplasm at the nuclear pore, but yet there are a few people who keep pushing this idea that the capsids are intact when they enter the nucleus. I mean, there are other explanations for it, which they...
A little gentler on the A. A little earlier. All right, run the other way.
So if I cross this, I will be...
This is the last boss.
This is the last boss?
If you beat him, the game's over.
Oh, man!
Turn around. Try it again. Tap B. And A on the way down. Like when you're at peak, press A. Oh, that was close. That was close too. A little earlier on A. All right, run away. Try it again. All right.
I see how easy it is for you, Ken, to tell me about this. Run away, try it again. I'm like, what? Dude, it's not shooting.
In my defense, this started out as a joke, and we make every guest do it. So you're going to be on the list of every guest we've had that's played this game.
Oh, man.
And the time you've taken to beat him.
Yeah.
It is absurd, though, is it not?
Yeah, Mr. Vegas. That's too crazy.
We could probably see these capsids with a DIC at 40,000 times. That's
I've never tried it out, but
do you know if we've taken any videos of that living blood doing its thing? Can you see it with an optical microscope?
yeah, what living blood, white blood cells, how they move and how they divide, we can see all that while optical microscope. But seeing them simultaneously as the virus moves into the nucleus or in the cytoplasm, you know, the speed at which virus travels, they travel about 2 micrometers per second.
Oh, their comment was, no, I mean AIDS in the living blood.
AIDS,
Could you see
HIV in the living blood? Yeah, people have done that. Yeah,
Yeah.
as in like parts of the virus. And that's, for example, there was an image that you oh, Mr. Big,
The capsid image. Yeah.
yeah, come on, man, why are you not jumping?
It's hard. Also, if you guys have suggestions on who we should raid, we're coming up to the end of the stream whenever Francis beats Mr. Big and wins the war on drugs. We will be done for the night, but we're going to raid somebody else because we've had 2 raids tonight, which has been a lot of fun. So yeah, Draw Curiosity and Actual Education both raided us. You guys should check out their streams, education-related streamers. Small community on Twitch, but nonetheless, we love it. So yeah, run away. And if you get sick of this, I can intervene. Contra pulling out.
Yeah, come on.
We have an OSHA emote, actually. All right, turn around. Try it again. Gentle on A. Yeah, there you go. Little lighter on A. So So like right when
What is this light and not light only? I don't understand.
so the way they made this work, they have two different buttons, right? They had to turn into one. And so they said if you touch it in the first 7 frames, rocket. If you go beyond 7 frames, it's a bullet.
That's
That's how they designed this game.
That's so funny. Okay, I gotta get him,
It'd
man.
be easier if we had 4 buttons.
Yeah. Oh, I got him.
Yep. And so it's that timing is everything in this. So your jump is good.
I'm just learning to jump. You're like a baby. Hold on, let me see.
It's amazing skill.
Yeah, to touch it just as good. I mean, I could pipe it. I could use a 200 micron pipette.
Exactly.
And
Like
pipe it.
nanomole concentrations.
Yeah, I can do all that. But the jump has been so difficult. This guy is smiling at me. Look at his teeth. Oh, jeez, man.
And he's shooting tongues.
Yeah,
at
this is
This is biology at its finest.
Oh, man.
It's very war on drugs with the impotent controls. Absolutely. This is a metaphor for the actual war on drugs. We cannot control it. So what do you think the world record time on this game?
I don't know.
So you're at 28 minutes and 50 seconds.
Okay, what's the world record time on this guy?
9 minutes and 4
Oh
seconds.
no! So it takes longer to play this than Mario?
Yes.
I don't believe it. Oh
It does.
jeez.
And I know, because I am the former world record. My time is 9 minutes and 7 seconds. So I'm the second fastest at this game.
So how did you figure out that you have to shoot him in the head?
It's just trial and error.
Yeah, it's
a
right.
lot of trial and error. There's actually a very particular way you beat this boss. All right, stop, turn around, tap B. There you go. Lighter on A. Lighter on B.
Oh, man.
So don't even push it down all the way. Just like touch the button so it just needs to make a little electrical contact. Yeah. You can even do piano touches. Like put it down and oh, there we go. That might be the way to do it. A little earlier on A. Oh, that was good. I like that.
Okay.
We've never tried the piano gameplay.
Yeah, this is easier. My thumb is
But who should we raid? Somebody put a name in chat. I don't know who's actually on right now. It's education related. Mahoodles, as usual, on a Wednesday night. She has to be West Coast because she's on regularly. Who streams on a Wednesday night, honestly? Oh, that was good. That was very close. All right, run away. Try it again.
his face and the way he turns around. So irritating, this guy is.
Turn around. Try it again.
Mr. Big.
So a little later on the A tap. Oh, on the way down. Oh, that was so close to the hat. But that's what you want to do. You saw how it went right over his head. A little bit later, and it's there. Yeah. So your options are: you do this, or I do this. If I do it,
what is the?
turn around.
What is he spitting at me?
That's his tongue.
Tongue?
That's his tongue. Look at it.
Why is he spitting his tongue?
I don't know. I don't know how you.
Oh, it's basically like, what is it supposed to?
Say no to CRISPR, kids. Yeah, where was this when you were a kid? You'd feed him like a drink and he becomes a giant head. We never figured out what show that was. The giant white dog.
Yeah, I'll tell you. Just as soon as I.
Oh man, a little too hard on the A. I'm gonna practice your piano skills. There you go. So you can see how gentle it is. Turn around, B. Close. Now it's just timing. I think you got the hits now. Now you got to walk away. Because if you do it when you're next to him, it doesn't do anything. You have to be a distance away for some reason, because Nintendo is dumb.
So how many tiles of distance at least you know? Yeah.
I don't know. All right, turn around. Later. Close, close. All right, walk the other way.
All right, guys, let's do this.
Better or worse than soccer?
This is so much better.
A little gentler on the A. All right, walk away. Try it again.
You know this, you got shot by this guy for a long time. All right, let's see.
So I had this game as a kid and I hated it. And then as an adult, I played it on stream when I was like the guest. And it became a running joke, like, you should try to beat it, and also you should make your guest play it. So that's why.
So 32 minutes is what I'm going at? Oh, geez, I'm over
you're
time.
32 minutes, but you're powering through.
Go for it.
All right.
Show me how it's
show
done.
you. So it's just really gentle tap with the timing. See how that went?
Yeah, almost.
It's very. I've not played this in a while. It's a very gentle.
Why is he throwing his tongue at you?
Okay, there we go.
25,000.
So now his glass is fixed.
Oh, the glass is gone. That's right.
Now
Oh, his head. Yeah.
Okay, so now you're
He's chasing you like a cow.
okay. So now you're gonna shoot bullets at him.
Okay.
And what you're gonna do is you'll press start, hold A, stand between these lines, and just move up and down, and then you can shoot him. Yep. See how the vertebrae went away and you got points. So run away from him. It doesn't shoot anything anymore.
Yeah, this is Mr.
So run
Big.
away, come down here, shoot bullets at him. Right, yeah, hold A. So you're gonna, after you die, okay, go down here, stay between these two black lines. Turn back, shoot bullets.
I want to be away
So
from
hold A, move up and down. Don't run towards him. Keep a distance. Contra, thank you for the support. Last minute questions, you guys. We're coming close to the end. We are about to beat NARC and win the war on drugs. So hold A, move up and down a little bit while shooting him, and then run back the other way. Do the same thing.
so irritating, man. It's the most irritating game ever. Jeez.
I'm glad you're okay, go up, shoot him more. Yep, and run away. Do it again, because you have to be in here to shoot him for some reason, so keep going. Yeah, so go down a little bit, turn around, shoot A, don't run towards him, just hold A.
Don't.
Yeah, run away.
Oh, you told me to run away. This is the biggest, so irritating, man. What the hell is he doing?
Still better than soccer.
Can
Turn around, shoot him.
I
Hold A, bullets, move up and down a little bit.
Can I not jump around and all that?
No, it doesn't help you on this boss. It's so dumb. This is Nintendo hitboxes for the win. Oh man. I think I
So
got
how long did it take to figure this out?
it took me a couple weeks of playing it. I figured out how to despawn him off screen and do all sorts of tricks with him just accidentally, but yeah. So go away, move up a little bit.
So just imagine, think about it, right? So before you
shoot,
before you would just figure this out. Now with the internet and all that, everybody knows.
Oh, yeah, no,
somebody figured it out. They let you know about this.
but they didn't, right?
Yeah.
Like no one beat this game fast.
Oh, is that right? Yeah.
Now there's a YouTube tutorial that I made that makes it.
You made it. You did.
Yeah, I did. I taught people how to beat this game.
Guys, if you're being so frustrated with this Mr. Big, I'll tell you how to do it.
Cut a puppy. It's over. Drugs have won the war on drugs. So yeah, go back up between these lines.
All right.
And so it's like, okay, so when he gets close, shoot A. Then there you go. You got two. Okay, now we're down to the last two. You got this. So run away this way. Turn back. Shoot him.
How do I run this way and shoot that way?
So just turn back. You don't have to run towards him. Just like that. Move down. Perfect.
Whoa.
There we go.
I done it.
We did it. We did it, ladies and gentlemen.
Jeez, guys, good. Okay,
No.
dude, awesome. This guy.
One more level. Keep going.
What? I gotta go one more level.
Civil forfeiture the level.
Oh, this is for all the gold. I'm collecting all the
You
gold.
don't have to collect the gold. It's taking time.
What's the
but
point? They killed me so many
points.
times. Yeah.
You know, you want the gold. Okay. It's taking time,
Yeah.
but totally worth
I'll
it.
take all his money. I mean, like this, Mr.
All right. Who are we rating? If anyone has suggestions, throw them in chat. Oh, that's
That's
the
it?
end. Yep. So that was 30.
Whoa, guys! I finished one whole game. I'm so excited.
No, you won a soccer game.
Yeah, one game of 3.
You won the war on drugs.
Yeah, and I went to 4 levels on 4 levels, 2 levels.
Well,
Or 4 levels on
you went up to
Mario.
2-3, I think. Or 2-2.
2-3, yeah.
But you get to put your initials in.
Ah, that's nice. How do I do
It'll
that?
be immortalized until I turn off this computer.
I see. So I just did this and say, okay, okay. All right.
Thanks so much for talking with us, Dr. Francis Contra. Thank you.
Always
for
such a
joining us.
pleasure.
Always, yeah, the gameplay is always a great time, but also the questions really carry us through. So we really appreciate it. Press end with the mutated finger. And then there you are.
I am not. Is that real or you rigged it?
It doesn't, it starts over every time.
Okay.
So congratulations, you did it. You won the war on drugs. Totally worth it.
That's awesome.
Oh, all right. 3 hours up, 3 hours down. We drank a 6-pack of Proof La La Land. We played some soccer. We played some Mortal Kombat, Street Fighter, Mrs. PacMan. We went through NARC. We won the war on drugs. How you feeling, Francis?
I'm feeling great. We talked a lot about science and stuff, and
Yeah, capsid.
this is my first time to be on a public platform like this. This is awesome. Yeah.
No, it's a fun venue, like the crowd. You saw the
Yeah.
questions. They're all over the place, but thank you. And any parting words for the audience? Anything you want to share? If you had a voice to the entirety of the world, what would you say?
I would say get on Amazon and give us those subscriptions.
That's right. Attach your Amazon Prime to your Twitch account and smash that subscribe button.
That's right.
Is there anything you want to say about viruses?
Viruses are cool. They teach us a lot about cells, and they teach us a lot about how to develop drugs and how to use them as therapeutics and stuff. I love studying viruses, and they're very cool. Yeah.
Awesome. No, it's been a fun night. I learned a lot. I really appreciate. I'm not a biologist, but I love learning about the field. So I thank you very much, Francis, again for joining me.
Thank you, Ken. This was awesome. Yeah.
Francis hasn't played games for like 20 years.
I know.
He was very nervous coming into it, but it turned out well.
It turned out really well. I
So
kicked Mr. Big's ass. That's good.
that's all that matters. So thank you all for joining us. As usual, we really appreciate it. Your questions really bring the conversation along, and we just love having those questions. Like it inspires ideas and thoughts and answers and whatnot. But yeah, so our next guest in 2 weeks on April 30th, it'll be Chris Skoberg, and he's an expert. He's a data scientist at Florida State University. He does data analytics for the university in terms of ranking and things like that, but he's also a sports analytics expert, which is kind of fun. So he's going to talk about big data processing and particularly related to sports, as well as university-based statistics. So yeah, I saw Chris today earlier, and he's excited to come on. We're going to see what he's going to play. He might actually bring his PS5 over, so he might play some modern video games.
Nice.
But we're going to see what happens on that. So yeah, swing by in 2 weeks. April 30th, we'll have another stream. In the meantime, again, it's always been a pleasure. Thank you for joining us for 3 hours of gaming and drinking and science. We always have a great time. So, yeah, stick around. We're going to raid some kind of education-related streamer, but we'll figure out who that is in a second. But yeah, always a pleasure. Thank you for joining us. Smash that follow and subscribe button and get notifications about future streams, because we will see you guys again in 2 weeks.